important to have time for at least a couple of questions. And most importantly, it's important to have time to have coffee and discuss things. So let's try to stick in time. So I will, or we will stand up after 10 minutes have passed, and that means you have 2 minutes extra if, for the presentation, and then there will be 3 minutes for questions. So.
Okay, so we are going to give the floor for the first speaker. It's Uwe Rascher for Forschungszentrum Jülich, who is going to talk us about the last 20 years of research in fluorescence and highlighting the current knowledge, uncertainties and knowledge gap towards the mission. So thanks a lot, Uwe.
Thank you, Martin. And good morning, everybody. I just realized that I'm the second day where I'm standing here as the first speaker in front of you. So good morning. Nice to see you back after hopefully inspiring discussions, which I at least had yesterday evening during the poster session and the icebreaker. So welcome to day number 2. And the first session is about vegetation stress. And when Mapi introduced me, I would like to focus actually really on vegetation stress, and I'm not sure if I really can cover 20 years, which I promised in my talk, but I would like to go a little bit back in the past years. What have we learned in the past, maybe 10 years, about the potential of SIF as an indicator for vegetation stress? And if I go back on one of the first figures which we used when we proposed FLEX 20 years ago, this was one figure which was in a lot of the presentations and also in the report for mission selection. And we argued at this time that photosynthesis is a central pathway which is important to feed photosynthesis, plant productivity, to finally have plant products. But on the other hand, we also said photosynthesis is affected by external threats, and we named there 4 main stressors. And those 4 main stressors were named because we had a reason to focus on these ones. There was a meta-study at this time, mainly on laboratory and small-scale experiments, which indicated that for drought, extreme temperatures, biotic stress, and nutrient imbalance, SIF may have the potential as an early stress indicator. And this is what I would like to go through. Was this true? Is SIF really able to measure those 4 stressors? And conceptually, measuring the stress, I think, is really important and linked if you also want to predict carbon uptake in actual photosynthesis or GPP. Because if I take this graph from a paper from, also I stole this from Nick, who's also in the audience, if you think about NPP or GPP, the reaction of stress, of vegetation stress, to an environmental stressor is important, and we don't know exactly what is the time dynamic, how stress affects photosynthesis. Are we in an early phase where stress may there but not affect carbon uptake? Are we somewhere in the transition phase, or are we already at the point of no return where finally vegetation dies? So vegetation stress detection is important also for carbon modeling. If photosynthesis is really happy, then normally we can work with potential maximum photosynthetic rates. But as soon as we have limitations, stress detection is important. And as a plant physiologist, an old dogma is a plant almost in natural condition never is happy. There's always some stress, always some limitation on the plant performance, which is pretty much stressful event is the normal, is normality. Just to recall you, this is my conceptual model, which I always have in mind when we think about how photosynthesis, efficiency of photosynthesis is related to the efficiency of fluorescence. We have this 3-phase behavior of the happy plant with high photosynthetic rate and a negative correlation with fluorescence emission. The second part, where the dynamic regulation with NPQ turns this relation in the other direction. And we have the severe stress of photoreduction, where all the dynamic regulation is not able to regulate photosynthetic and non-photosynthetic energy dissipation anymore. And then we have this great increase from fluorescence at the severe part. And if we talk about stress, we often talk about the transition between this red and this green part. Regulatory, still, the plant is still able to regulate, or not anymore. And then, with this, I would like to go to the 4 stressors. Drought is the first one. And I would like to refer to a paper which we did together with a co-author or head author of Alexander Damm in Italy in the preparation of the FLEX campaign, where we exposed an agricultural canopy to controlled drought. So we stopped watering in Italy, and then within days, the plants are faced to reduced water availability in the soil. And if you look in the classical remote sensing, we see that after about 1 week, maybe a little bit less, the plants started to wilt. A greenness index, in this case MTCI, was reduced. However, if we look on the solar-induced fluorescence on the 2 peaks, we already see a response earlier. Before, actually, I can see something in the greenness. I see a response earlier in the SIF signal. And this, we interpreted that this is the early reaction, the dynamic reaction of the plant to stress. The stomats close, the plants still remain wonderfully green. And then only later, we see the pigment breakdown, which we see in both parameters, in SIF, but also in the vegetation indices. So the 2 different elements, or the 2 different mechanisms, dynamic stress regulation because of stomata closure, can be seen in SIF, while then pigment breakdown can be seen in both, in SIF and in the other parameters. And this is a concept which after this we further rolled out, and I would like to highlight a study which we did together mainly with Juan Queiroz from my group, where we wanted to go from this 1 plot results to a larger scale, and we correlated the available plant available water in the soil by including some modeling of soil water with the emission of fluorescence. So it's a pretty extensive study over several years, over a larger area with several crops. And in this study, we could also show that actually there really a drop of plant available water, which is heterogeneous in the field and also different between fields, that this could also be detected in the SIF signal and more sensitively in the normalized SIF signal. So that's a new study just published. If you have fun, I recommend reading it. So that's drought. I think with drought, we have a good means of understanding to detect drought or to use SIF as an additional information in which phase the drought response currently is. Second stressor, extreme temperatures. Extreme temperatures is different if you talk about high or low temperatures. And one of the studies which I like a lot is the study from David Martini, which he did in Majadas, where he could show that at a summer extreme heat wave, he could observe actually the shift in the relation, or this tipping point in the relation between photosynthetic efficiency and fluorescence emission in a FLOX, in the FLOX data, looking on the grassland in the Dehesa, and he could see exactly this shift or this flip in the 2 domains between the regulatory domain and the non-regulatory domain anymore, where the ecosystem was kicked out of the domain where active or dynamic regulation was still possible. So this was high temperature. And I would also refer to some more papers where people looked really on heat waves. Georg Wohlfahrt is in the audience, having published a nice paper on this. So interact on this. I think we also have a lot of evidence that also in extreme temperatures, high temperatures, SIF seems to be sensitive. However, I would postulate that we do not know exactly what are the mechanisms, the underlying mechanisms behind there. And this is a domain where we went back in the past years more to the mechanistic studies in the greenhouse or in the laboratory or just in the field, where we used genetically modified Arabidopsis thaliana plants. This is the mouse of plant biologists. And I'm a plant biologist from our background, so we can use those plants. And they have a very specific knockout in some mechanisms of the photosynthetic machinery. And if you use these plants, they look totally the same as the normal wild type, but we know that internally how they shift their electrons, they have a limitation somewhere on a really controlled and well-defined matter. Kevin Aspon already came forward with a model. What happens, for example, if we expose a plant at highlight during a winter cold spell? This can happen in the spring if suddenly the plants are already getting greenish, but suddenly you have a cold spell in the early spring. And we know, and we have a hypothesis, how actually energy is shifted and how this is linked to the fluorescence yield. And this is something which we are currently trying to understand a little bit better by a new ground-based imaging system, which Huayu Peng used during her PhD thesis, where we again scanned these Arabidopsis thaliana plants, different genotypes. Those are genotypes, this is the normal one, and where we could see which element of the plant is affected by this drought spell, or, that's what you did, also a longer lasting drought during winter acclimation. So what I wanted to say, extreme temperatures. From the plant physiological side, we know quite a lot what is actually happening in the plant. We have some understanding how we can link this to SIF. However, I think we are still lacking the really link of this information to then long-term ecosystem studies where we monitor SIF during winter, during extreme temperatures. Let it be high temperatures or let it be low temperatures. There, I'm at the point where I would like to point out one important thing. I was now always talking about the efficiency of fluorescence. And if I talk about efficiency of fluorescence, as a plant physiologist, ideally I would like to know what is really the efficiency of fluorescence emission on the leaf level. And that's a big challenge, which is still an open challenge, like how do we come from the 300 meter pixel to the mechanisms happening on single leaves. We have there some empirical methods in hand in the meantime, because we know that if we have, for example, an extreme environmental condition, some stress, it's not only the physiology which is affected by a plant, but also the structure of a plant is greatly affected. I think we have the three tools to handle this. On the one hand, in the meantime, we have the so-called structural functional plant models in hand, which are having greatly advanced in the past years, where we can use really a full understanding of what happens structurally and functionally in the plant to scale this up to the satellite. We also have the radiative transfer models, which we can invert, where we have where we can represent a three-dimensional canopy in more detail and think about how fluorescence is actually reabsorbed in the canopy, but we also have these empirical models or the empirical approaches in it. All are not possible. However, we somehow need them to really go down to the regulation where photosynthesis happens. Last 2 minutes, I want to speak about two challenging things which we have not covered fully, and this is, for example, pests and diseases. And while I have said with drought and with temperatures, I'm kind of optimistic that we can handle this. At least all the leaves in a big canopy are normally affected, or we can assume that all leaves are affected by drought or extreme temperatures. With diseases, it's different. If we talk about diseases, we are talking about a few single leaves where one leaf tip may be affected and show some sign for diseases. Just a few pictures how this looks. Additionally, diseases are really complex, hundreds and thousands of diseases, and even for the good farmer, it's really difficult to separate which disease is actually affecting my field. So diseases are really complex. We always, at the moment, I would postpone, we need a closer look. So we need really a close range observation of the diseases to be really specific there. We have to combine this most likely with modeling and more data. There's a review paper which I wrote with Ann-Kathrin Berlein. And if we think on the larger scale, we had a long workshop on the Plant Pathological Society approximately a year ago. And we thought about, can we detect diseases from a 300-meter satellite with 300-meter pixels? And I went out with the thing, well, most likely not. 300-meter pixels are really too coarse to really see early or specifically a disease. However, the disease community has this big possibility of really epidemiological modeling where they can predict disease spread in a really sophisticated way. And I would say that's the way where we can actually work together or link together. And the last thing, and then I promise I'm at the end, nutrient imbalance, I think, is even more complex, and I would make it really simple. This picture in the audience, who can tell me if this is a nutrient limitation or a disease? I would say it's almost impossible. And also, if you put this on a plant person under the nose, we don't know. It can be both. It could be nutrient, it could be phosphate, it could be a disease. So for nutrients, I think we are really even on an earlier step. The good thing, or what I currently say, is nutrients mainly affect plant pigmentation, and there I would first look on the classical optical remote sensing. So with this, the summary, what do I want to give you for this, for the thinking? I think fluorescence is a good tool to add crucial information on what's happening really inside the plant, also for stress reduction. We, however, have to combine this with the other information. Thermal for drought detection, optical remote sensing, potential information on structure. There are 2 papers on this topic where we try to elaborate a little bit about what sensors and how to integrate different sensors with different time resolution, different optical domains for stress detection. And with this, just as a summary, so yes, drought, I think we are already pretty far and we can do quite something. Also for extreme temperatures, I think by combining SIF with optical remote sensing and temperature is a good way forward. And for nutrient and diseases, I think we still have quite some challenges, which I tried to put together or summarize here. So with this, how badly am I over time? Sorry for this, and I thank you for your attention.
Is there any question in the audience? So I have a question, Uwe. Sorry. So you talk about this non-linear pattern between photochemical yield and fluorescent yield. What we can do to characterize that? So I'm thinking, so we have MODIS, and to predict GPP, they have characterized different biomes to improve the light efficiency coefficient. So can we do something similar with SIF?
Yeah, that's something which I really also scrap my hands over. Where in the scheme on this red or the yellow part, how do you know in which domain are you actually handling? We had the idea with FLEX that we can actually measure NPQ directly. This was one of the main ideas to say, well, can we determine the degree of NPQ? Can we determine, are we still in a healthy range of NPQ regulation or not? However, NPQ detection from a satellite is also really complex. You saw this yesterday, I think, in the talk from Christian van der Tol. He mentioned this in his talk. So I would go exactly what you suggested, use additional information, maybe also from other satellites, potentially also for modeling, to determine in which domain we may actually falling at the moment when we do our observation. And I'm totally with you, most likely we will need also other information or also climate modeling for this.
Okay, thank you, Uwe, again. And let's move to the next speaker, so Sebastian Wienecke, and from the University of Leipzig and German Center for Integrative Biodiversity Research. So we'll now hear how important it is to consider the variation in species and also the time of the day when it comes to interpreting data. So, floor is yours.
Okay, I think, yeah, now it works. So it's the last days. We already talked about some of the key challenges, so I will not go deeply in repeating them. It's just like what I would like to mention is the nice thing about fluorescence is we can measure it at really different kind of scales, and from each measurement, a different scale, we can learn something new and to explore the signal from a different kind of perspective. So what I would like to concentrate on my talk today is the complex SIF photosynthesis relationship, and that's basically what Uwe mentioned in his talk about this relatively complex relationship between the yields of photosynthesis and the yields of fluorescence. And this is a figure I also always have in mind when I look at fluorescence measurements from José Moreno from ESA ESRIN, which basically also based on the van der Tol paper, where we see what also Uwe already explained, that we have like this different kind of stages where we see that fluorescence under non-stress conditions have a negative relationship to the yields of photosynthesis, and then with increasing non-photochemical quenching we are on a positive slope. So the question is, this makes everything really difficult to interpret the signal because we really need to know where are we. So under extreme stress conditions, we can then also see that we are going on a negative slope again when fluorescence is increasing while the photochemical yields are actually decreasing. So what we try to answer in our in this work I want to present is how the relationship between the quantum yields of fluorescence and photosynthesis are changing over day, season, and we have measurements over 2 different kinds of tree species which have very different kind of physiological reaction to stress. So we wanted to see, can we see actually like this kind of behavior? So we set up an experiment in 2022 and we measured over Fagus sylvatica and Tilia cordata. Fagus sylvatica is European beech and Tilia cordata is small-leaved lime or small-leaved linden. And we choose these 2 specific trees because they have different kind of reactions to stress. So the Tilia cordata has a lot of leaf angle movements under stress because the turgor pressure is really can get very low, and then you see a lot of leaf movement under stress conditions. And also is an isohydric behavior, so it closes the stomata much closer than the Fagus sylvatica. And so we measured fluorescence with the FLOX system over these 2 trees. And at the same time, we had micropumps installed within the canopy to get information about the photosynthetic parameters. And at the same time, we had a lot of environmental measurements, which are really important to know, okay, under what stress conditions are the plant actually. So in 2022, we had like measurements, or this campaign lasted for around 100 days, and we had observations like every 15 minutes from the micropumps, and then we could like see like, okay, how is basically like the seasonal and the diurnal relationship between these parameters. So the first thing what we want to do, and that's probably something you already see like plenty of times, we can relate photosynthesis or electron transport rate to the 2 peaks of fluorescence, and these are fluorescence values without any correction, and we can already see, okay, we get quite nice relationships, but we also know that these relationships are mainly driven by the amount of light. And we also need to correct for the escape probability, because on these 2 peaks we have 2 different kind of mechanisms which really define how much photons or how much light we actually receive at the sensor. And for this we need to estimate the escape probability in the red and in the far red. To do so, there's also, Uwe mentioned this, there's a different kind of empirical algorithms. We could also use like models like SCOPE or DART to get to these values. For the escape probability in the far red, we have, or I tested 3 different ones. I will not go into the details, but it's basically like a near V-based approach, the FCVI, or a new approach, MSC from Key 2025. And we then compared like the metrics to see like, okay, how do they actually impact the interpretation of the signal. And here's always a challenge. So which model for F-escape is actually the best? And that's really tricky because we can't measure the escape probability. So we have to rely on a different kind of strategy. So we can either look at some metrics to compare like, okay, how does it actually, the relationship change? Or we use models to calculate the escape probability, but then it's always a question, do I want to set up a really complex model like DART, or how much can SCOPE actually represent the canopy I have? So what we did here is we compared the metrics, and what we would expect is that we have a higher correlation, or higher correlations is the better escape probability model, which is also not completely true, because if we go actually into stress, we have a decoupling between APAR and fluorescence. So it's then the challenge, if we have a decoupling, a better relationship does not necessarily mean we have a better model for escape probability. So we did this, and here for the far red, we actually see that all of the models perform very similar. And also we see that for the lime tree, we had like much lower performance for the electron transport rate relationships than for the beech. Then we have like 3 models for the for the red. And here this is a little bit tricky, but why is it tricky? Because we're still struggling to figure out what's the best way to account for the reabsorption effects. But at the same time, the red fluorescence actually relates much better to the physiology of the plants. And I will show this later in the next slide. So here we selected the near V-based approach because we basically saw the best metrics. And also when we looked into the relationship, they made the most sense. And I will come to this now. So if we want to get actually information about or relate fluorescence to the physiology of the plant, we have to look at the yields. So it's not enough to look at the absolute values because it's driven by par. So the yields are basically normalized by the amount of light. And if we look for our measurements, or we compare the red fluorescence and the far red fluorescence, we see that the red fluorescence actually contain a much stronger relationship to the measurements if we look the daily mean. But at the same time, if we like compare all the measurements, like every 15-minute steps, we can see that in this heat density maps that the relationships are much more complex. And even worse for the far red fluorescence. So why is this so complex? Because of different kind of diurnal changes over the season and different kind of stress conditions, which also have a strong impact then on how we interpret these relationships. And in the end, if we do not consider that, we end up with big clouds of data points, which are very hard to interpret. So to better make sense out of it, we had to group the data into different kind of stress groups. We did this by running a PCR on the environmental parameters and then use the first 3 components, which explain 90% of the variation, to as an input for a k-means clustering. We identified 4 groups, and then we selected the 4th group, which selects the most stressed days, and then had like basically 5 different groups with different kind of stress conditions, and we can categorize them if we then look and like, okay, how are they basically, what kind of environmental conditions do they represent? We have the different kind of stress groups where they represent first the light conditions with gray sky for the first diffuse and clear sky then for the 3rd, 4th and 5th. And if we look in the atmospheric dryness and the soil dryness, then we can also see like, okay, what were basically the water availability to the plants. So, and in the end, we have a nice gradient in the photosynthetic yields to basically look, okay, what are the stress conditions for this campaign? We then grouped like all our measurement days into this stress groups, and then calculated the mean. So this is what you can see here. Why did we calculate the mean? Because first we wanted to reduce the uncertainty, and we wanted to look for general patterns under the certain kind of stress conditions. And then afterwards I used a filter, a Whittaker filter, to get like a general relationship to this. I have to unfortunately speed up. So this is basically what I did. I will not go into the interpretation of this. If you're interested in it, we can look at it later. But if we do so, and use basically like this, the smoothing filters for the phagosylvatica, we basically were able to derive this kind of slopes for the different kind of stress groups, which basically show this is like the morning hours, where we are under light limitations for like this is for stress group 1 and stress group 2, 3, 4, 5. And we see that we have like a seasonal behavior or like a stress related behavior. At the same time, also this diurnal mean behavior, where we nicely actually see that we are here under light limitations, then we are on the positive slope, and then we are basically have the hysteresis where we have again a negative slope again. So for the telia, it looks a little bit different because the telia already regulated much faster the stomata closure. So we already started with our measurements on the positive slope. And then we did not have like this shift we had here, or like this long hysteresis, because it immediately started after solar noon that the recovery started. So what does this basically mean? If we print all of these behaviors into the whole plot, we see it looks quite messy. And here I indicated the overpass time at 10:30 and the overpass time at 1:30, so like flex and tropomi around overpass times. And then I calculated, okay, if we only use this overpass time data points, and then check how well it works to actually relate it to our measurements, we see it gets really challenging. For the phagus, for example, if we only look at the stress groups where actually we have light conditions which would be observed by a satellite, yeah, it's basically a cloud. It looks much better for the telia, where we get like higher correlations. But still, it is like the question, okay, if we have two different species who react very different, this might be a big challenge. Tropomi, similar problem. And then what I did, I actually calculated only the data which are on the positive slopes from both plants to see how, if we really know where we are on the slope, and if we can only select the data where you're on a positive slope, we can overcome this challenge. But for this, we really need the diurnal information or need to know where the breaking points are, where we are then on the positive slope. Okay, so as a summary, red versus far red, in my experience, I always, if I want to relate to physiology, I have much better experience with the red. The far red often delivers very strange results I cannot understand, and I'm not very happy with the far red. So if you don't work with the far red, only with the red and only with the far red, try it. It's worth it. And then the nonlinear dynamics. These are really challenging aspects, especially if we have like species who react differently, because for remote sensing implications like from satellite, this would mean that that a single overpass would probably not capture this dynamics, and then we are basically miss predictions. At the same time, if we average or make a spatial average of a different kind of plant functional types or plant functions which react differently to stress, this might actually average out, and we do not see these clear signals we would like to have. So what could be possible solutions? We need to think about sensor integration. So is there a possibility to get diurnal cycles from geostationary platforms? Can we use high resolution for functional diversity maps to actually have an idea, okay, which plants are under react differently? And of course, FLEX, which is the only satellite which will deliver red fluorescence to close the temporal spatial gaps. Thank you very much.
Thank you, Sebastian. So we have time for a very quick question. Yeah.
Thanks for your presentation. So very quickly, what do you think, how many observations per day do we need to adequately sample the diurnal cycle to get the information we require?
Yes, so that's a very challenging question, because it really depends on also what plant or like how the plants react, and we need to know that. So the most challenging aspect is if we are measuring like in this region where it becomes really hard to know if we are on the positive or negative slope. I can't give an ultimative answer. It would be good to have something in the morning, in the afternoon, and in the late afternoon. That would probably give like a bit of more information. But still, can we really get the breaking points? I think the breaking points are really the goal, should be the goal.
Okay, thank you very much.
Thank you.
Thank you, Sebastian. So we move to the next presenter is Carolina Sakovka from the Italian National Research Council. She's going to show us the results of the last campaign regarding the FLEX-Calvat validation and also a study underwater stress. So thank you, Carolina.
Hi to everybody. Yeah, so FLEX-ITA is an Italian project funded by the Italian Space Agency, and its main objective is to support the FLEX mission and to enhance the scientific exploitation of the SIF airborne data collected over vegetation and aquatic ecosystems. FLEX-ITA consists of a few working packages, including one dedicated to installation, setup, and acquisition of airborne data. Another one focusing on the development of SIF retrieval algorithm, and the other two dedicated to measurement campaigns over vegetation and aquatic ecosystems, respectively. We already completed all field campaigns within the FLEX-ITA, but today I would like to focus on the vegetation component of FLEX-ITA and the last summer data. But if you're interested to know more about SIF and phytoplankton in Lake Garda, I invite you to look at the dedicated poster. The main airborne system within FLEX-ITA is the IBIS, SPECIMS IBIS camera. However, due to the collaboration with Forschungszentrum Jülich, to compare hyperspectral data for SIF retrieval coming from the two systems, IBIS camera was flown in tandem together with the well-known HyPlant system, both in 2024 and 2025. In 2025, the airborne acquisitions took place in Grosseto, Tuscany, Italy, over 3 days, 13th, 14th, and 18th of June. In 2025, FLEX-ITA operated in synergy with the FRM for Fluo project, and thanks to this, the airborne data are going to be enriched by remote sensing data coming from a helicopter, drones, as well as from proximal sensing SIF data coming from FLOX systems which were deployed across various crop types. So basically in 2025, the area of Grosseto was pretty extensively investigated, providing a unique multi-scale SIF data set. Within this presentation, the SIF processing method is SFM, and within the processing chain, the atmospheric correction is performed efficiently, calibrating the H1 parameter in MOTRAM using non-fluorescence pixels. And this automatic procedure is being refined for IBIS. So for the time being, we use the H1 parameter derived from HyPlant and applied it to IBIS, and the results are good. The spectral patterns were reconstructed well, but of course the results are not optimal yet, or not fully consistent, considering different target surfaces, namely soils or dry vegetation versus green vegetation. When we compare the HyPlant SIF against ground-based FLOX SIF, we found a good agreement with points oscillating around the 1-to-1 line. We are also working on comparing the airborne data against the drone data. The first results are encouraging. However, more investigations are going to be done, taking advantage of an incredibly high number of coincident points, allowing for robust cross-platform comparisons. We also carried out a water stress experiment, as we're interested to know whether SIF provides an additional value for early detection of water stress compared to reflectance-based remote sensing methods. And in 2025, we were dealing with mice and tomato. And within those fields, we selected a parcel which was then divided into water-stressed and irrigated plots. Both fields were equipped with fluxes mounted on structures equipped with moving arm, allowing alternative observations of water-stressed and irrigated canopies. The irrigation was applied differently to water-stressed and irrigated plots. So irrigated plants received regular irrigation that followed local agricultural practices, whereas the water-stressed plot was subjected to controlled water limitations achieved by timely, strategically timed irrigation events. We measured a number of parameters, including leaf gas exchange with Chiras and Lycor 6800 equipped with PAM. And those measurements were carried out in the morning, midday, and afternoon, so 3 times per day, while other parameters like active fluorescence, leaf water potential, and leaf chlorophyll content were measured periodically during the field campaign. We also have near continuous canopy subflow data measured at half-an-hour intervals in both canopies. But let's move to some results. And here we are talking about mice and Chiras-4 data. So Chiras-4 showed clear differences between the irrigated and stressed leaves. Irrigated leaves maintained higher leaf photosynthesis, stomatal conductance, leaf transpiration rates, and ETR. Unfortunately, we don't have Chiras data on the 14th of June, which was our big stress day, due to sensor malfunctioning. But if we consider those data together with the Minipam dataset that was available on the 14th of June, it was confirmed that after nocturnal irrigation between 14th and 15th of June, mice leaves recovered partially. But then continued soil drying led to progressive stress, culminating on the 18th of June, when the physiological parameters reached the lowest values. And then nocturnal irrigation between 18th and 19th of June led to the second recovery phase. The story of tomato is slightly different. And here on the 13th and 14th of June, leaves were already stressed. Then the nocturnal irrigation applied between the 14th and 15th of June led to the recovery. But here in tomato, irrigation was continued on the 15th and 16th of June, which led to further recovery. Next, irrigation was suspended. And on the 18th of June, we saw the second decline of physiological parameters of water-stressed leaves. Finally, we applied the last nocturnal irrigation, which allowed to observe the second recovery of water-stressed leaves. We do have leaf water potential data supporting the previous results, as they were collected during the stress and recovery event on both crops. Also the canopy subflow data, here example from mice, see very well the recovery. Like here we talk about the 14th, 15th, the yellow line comes closer to the blue, and the same on the 18th and 19th. We haven't noticed differences in leaf chlorophyll content in the tomato. That was measured with the Dualex. However, the leaf samples collected in the fields have yet to be analyzed in the lab. Instead, water stress caused reduction in SPAD values in mice. Now, if we look at the FLOX data and tomato, here only the data from the 2 stress and recovery events are presented. What we see is that APAR was very similar between the water-stressed and irrigated tomato. But then if we take a closer look at the first stress and recovery event and the fluorescence yield, we see that water stress caused reduction in fluorescence yield on the 14th of June, which, considering the very similar APAR, suggests functional stress rather than structural. Then, after applying the irrigation overnight, what we see that APAR remained very similar, but the nocturnal irrigation brought the yield of the two treatments closer together, which suggests a recovery, physiological recovery of tomato plants. The situation in mice is more complicated because of stress-induced leaf rolling. And here, if we look at the first stress and recovery event, we see clear differences in APAR of water stress treatment across those 2 days, the same during the second stress and recovery event. So further work has to be done from our side to find the best way to normalize CIF, considering these effects, and just to visualize how strong this leaf rolling was. Here the photo of irrigated plot and water stress plot during the stress day. They look completely different, right? And then the same photos of irrigated and water stress plot after irrigating the plants. So the situation there is very dynamic. Thank you.
Thank you, Carolina. Is there any question?
It's just a silly question, but I keep on wondering about it. On your CIF maps, you show it also with negative values, and I see it all over. So I keep on wondering, why are we showing actually negative values in vegetation like LAI? That would not make sense, but is there any reason why CIF values, negative values are being shown on maps?
Yeah, the maps, the negative values were probably related to soil or senescent vegetation, but considering that Sergio Cogliatti is here and he's dealing with this, maybe he can answer you in more details why we do see this.
Any other questions? Yeah, please.
Thanks for a nice work. Regarding the last picture that you were showing with these roll-up leaves, that's really striking to see how the structure of the vegetation changes from one day to the next. Here, it's just a suggestion, because here probably the indices related to vegetation fraction will show up a change in the amount of vegetation, and you can use that to normalize that.
Yeah, the results are preliminary, and you are right, but the issue is that those leaf rolling is not only going to change from one day to another, but also at different times of the day, which makes things even more complicated. And yeah, we are playing around with different normalization methods, and at the moment, if I used APAR derived from FLOX, which is NDVI based, versus APAR derived from scope inversion, at the moment the results are not consistent. So there is still some work ahead of us to...
Yeah, but probably scope is not the best model to use here in
It
this
might, yeah.
really complex structure.
Okay, thank you. Sorry, I think you can continue if there is more questions in the coffee, and thanks, Carolina, for staying in time. So, Maria Pilar is very good at multitasking, and apart from chairing this session, she is also having a presentation. She will tell us about the nonlinear relationship between fluorescence and photosynthesis in response to very different types of stress, diving into.
Thank you, Albert. So good morning to everyone. So there had been several publications where they saw this linear relationship between fluorescence and GPP. Mostly these relationships are driven by long-term dynamics, which are linked to pigment pools. But as has been mentioned through the different presentations of this morning, we also see a nonlinear relationship, which are mainly driven by dynamic changes in the lab reactions, which give us information about short-term dynamics regarding par, temperature, water availability, and nutrient availability. And these are the ones that we can use to detect early stress. So based on this information, and also regarding the different light phases that Uwe was presenting before, so I come up with 3 different questions. So the first is how fluorescence can contribute to the linear and nonlinear models to estimate GPP under stress. The second is how growth light conditions and stress influence these shapes of the dynamics of these phases. And the third one is how prevailing environmental conditions affect the dynamics between fluorescence and MPQ under stress. Starting with the first questions, we have a WIT experiment where we have a WIT experiment in open field where the plants were growing under high light conditions, and we have 3 different treatments, low, medium, and high nitrogen. We did measure different parameters, all at leaf level. We used the fluobat to estimate the total fluorescent spectrum, so the total fluorescent shape, but we also measure reflectant absorbance. So we will be able to estimate the absorbance of different pigments using the Sullivan-Wittenberg approach. So we'll be able to determine the absorbed light by the chlorophyll A and also by different pigments related to photoprotection, so carotenoids and xanthophylls. Also because we have reflectance with the right different vegetation indices as NDVI, PRI, and the Reddit. But also simultaneously we measure with the Lycor 6400, so we will be able to derive ANET and also active fluorescence and derive photosynthetic yield. So just a brief introduction. So we use different models based on the Montiel light use efficiency, where we have, so basically on the equation of the light use efficiency, we use different parameters that I have defined as a proxies for light use efficiency to FAPAR. But also following the similar strategy, we look into nonlinear models, mostly look into Gaussian process regression models. My goal was not to determine, to create a model based on Gaussian regression models, was most to understand which of the parameters that we are measuring are providing more information about the variation of photosynthesis. From our study, what we see is that we don't have a linear relationship between light use efficiency and FQE. And this is why most of the linear models didn't provide us a good and accurate estimation of photosynthesis. But then this is because, as I say, we also measure in the field with active fluorescence, and when we see the relationship between photochemical yield and fluorescence, we see this nonlinear pattern that was shown before. And most important, because our plants were growing under high light conditions, most of our measurements were in this range. So when we apply a nonlinear model, we improve our estimation. Again, my goal is not to show that we can build a nonlinear model based on Gaussian regression processes, but for me the most important is to see that when we use a nonlinear model, the two parameters are more relevant or are giving us more information about the variation on ANet was FQE and the REDET. And importantly, the REDET information indicates a downregulation of the light harvesting processes for photoprotection, which could be linked to the sustained MPQ. So, and you will hear more about this in Adrián Moncholi's presentation, which is followed up in the next session. Okay, so the main message for this first study is that there is a need to build up nonlinear models to be able to use fluorescent information to improve the estimation of photosynthesis. So our next experiment is how growth light conditions are going to affect, how growth condition and stress are going to affect these dynamic shapes in the relationship between phototypical yield and fluorescent yield. So this is a paper that has been just submitted by Cynthia Chardy, who also have a poster here. What we did is, through different experiments, we have been measuring light curves of fluorescence. And specifically in this presentation, what I'm showing are 3 different experiments: tomato experiment, barley experiment, and corn experiment. Tomato was growing under low light conditions and have both nitrogen and water stress. Corn was grown under high light conditions under nitrogen stress, and barley was grown in a greenhouse under intermediate light conditions and also water stress. So as I say, we use the light curve to derive different light curves, and we derive the photosynthetic yield, fluorescent yield, and the total MPQ yield. So these are the main results. We have here corn, high light conditions, barley, intermediate light conditions, and tomato, low light conditions, and we are showing here the photochemical yield versus fluorescent yield, and then here MPQ yield, total MPQ versus fluorescence, and here MPQ versus photochemical yield. I'm going to focus in the first plots where we show the relationship between fluorescent yield and photochemical yield. So my questions here our questions are how the light condition and the stress is affecting the duration of these phases. So what we show here is the par, the accumulated par. So how much par needs to be accumulated to change from one phase to another. So the first thing that we observe is this is phase number 1, phase number 2, and then the last phase. That plants growing under low light conditions, that means that this change is going to happen earlier, so with lower par, which phase number 1 is with lower par, phase number 2 with higher par, which means that phase number 3 gets extended, so it's longer. And then regarding to the stress, we see that this also short, the stress will short the change from one phase to the other. And then it's just regarding what Sebastian was mentioning earlier. So this breaking point, so this the photochemical yield breaking point that you move to one phase to another. We also observe that when plants are growing under light conditions, this breaking point happens earlier, so under higher photochemical yield, mostly in phase number 2 and phase number 1 and 2. And then regarding stress, it's a similar behavior. So plants under stress, this breaking point happens also earlier, so under higher photochemical yield. Opposite trends were observed in fluorescent yield and MPQ yields, but I didn't have enough time to show it here, but you can see in Cynthia's posters. So the take message for this experiment will be that growth light conditions, or I will say in general environmental conditions, matter to determine these dynamics or the duration of these dynamics between photochemical yield and fluorescent yield. And then when it comes to the last question is how the prevailing environmental conditions affect this dynamic I was mentioning before. We have run 2 experiments at the University of Valencia. Also for the lack of time, I'm only focused on the first experiment. But at the University of Valencia, we have a growth chamber where we can modulate the environmental conditions, so light, temperature, humidity, But also, simultaneously, we can measure with different sensors, with active fluorescent, hyperspectral sensor, thermal sensor, and a 3D reconstruction camera. In this experiment, in this presentation, I'm going to only focus on the active fluorescence data, but we are still analyzing the passive fluorescence and the reflectance and temperature dataset. I say we have two experiments. The first one, what we did is to mimic the diurnal cycles and in tomato plants, and we grow then from the moment that we seed the plants in the growth chamber, so they will all grow under highlight conditions, and then we have control and drought. And the second experiment that you can see in Cynthia's poster, we keep the part constant and we just change the temperature, so we create a heat wave. So I say I'm focusing in active fluorescent measurement, so based on the recent paper of Albert group, next slide will be similar to you, Albert, because I just copy-paste what you did with our data. But we derive both for the length of the experiment and also daily, the photochemical yield, the total MPQ differentiated between sustained and reversible, and the fluorescent yield, as I say, for the length of the experiment and also daily. So what I'm showing here now is the results for the diurnal cycle experiment and for the complete experiment. So here is the control and here is the drought. And in green is the photochemical yield, in blue is the sustained MPQ, yellow is the fluorescent yield, and in red, brown is the thermal dissipation yield. And what we see here is that the experiment, through the experiment, the dynamics were modulated mostly by when the sustained MPQ appears, and in this experiment that happened at the 5th day of the experiment. When we come to the diurnal changes, we see that so here is before the stress, during the stress, at the recovery phase. Here we are representing the total MPQ, which is the sustained plus the reversible. Here is only the reversible and then the fluorescent yield versus the photochemical yield for the drought and control plant. So what I think that is really interesting of these results is that at the beginning, the dynamics were really similar, and that we are in the phase of a negative relationship between fluorescence and photochemical yield. And is here in this experiment is the total MPQ, which have information about the sustained MPQ, which is giving a difference between the different treatments. The reversible MPQ is not, doesn't provide this difference, and then fluorescence is also giving us this information between the different treatments. And then in the recovery phase, the total MPQ remains high, and also the fluorescence is quite low, but this is because the photochemical yield increased after the watering of the plants. So when it comes to the question of how we can use information to detect stress, I think that when a temporal series come important. So what I'm plotting here is the deltas between the different days. So it's 1 day minus the previous day for the total MPQ and also the fluorescence. And what we can see here is that there is a moment where the total MPQ increased, and that happened during several days, so it's an indicator that there is a stress there, so a change in the pattern, and also a significant change, and then fluorescence decrease. So studying these temporal dynamics and looking into this changing into the dynamics, I think that will be a good proxy for determining stress and early stress. And interesting is this day that we see this change in the dynamics. The results are not only significant when we look to the total, but also when we were measuring at each specific moment of times of the days. So the home message for this experiment is that the sustained MPQ and fluorescent yields emerged as the most sensitive indicator for stress, and that to be able to detect stress, we should look to the temporal series and then change in these consecutive changes in the dynamic between MPQ and also fluorescence. Just to finish. So the conclusion is that the nonlinear models matter, and we should invest new experiments or new modeling approaches to take into account these modelings, where both the sustained MPQ and the fluorescent are important, that the grow light environment conditions also matter, and I think this is why it's important to have fluxes or monopans in the calval sites so we can have these temporal dynamics and maybe, as I was saying before with Uwe, maybe parameterize regarding different biomes how these dynamics behave. And then to diagnose stress, both sustained MPQ and fluorescent will be important. And again, we should look for these temporal dynamics to see when we have these changes. So sorry for speaking fast. We will have time to talk later, and thanks a lot for your attention.
So thank you, Mappy. We have time perhaps for a very quick question. The audience? If not, I would say that maybe we move to the next one and then we stick then to time. So the next speaker is David Herrera from the Research Center Jülich, and will tell us about some advances about tracking soil drought with sieve. Very interesting topic.
Yeah, thank you very much. Yeah, I'm David Herrera, PhD student at Research Center Jülich in Uwe's group, and I'll be talking about a study called Tracing Changes in Subsurface Water Storage Through Novel Satellite-Based Time Series of far-red Solar-Induced Fluorescence Quantum Efficiency. And basically what this is I come from a different perspective to the talks we had before. I come from the satellite perspective this time and not from the ground. And what we did is we combined troposive observations and tried to find a connection between drought stress, basically. All right, so why are we interested in the vegetation response to water stress? In this figure by Alex Damm, you see in the bottom the water availability from left to right, from high to low, and then the timescale of adaptation processes from very fast, so microseconds to months. And then in the top, you have the various satellite products that we have available. So the fluorescence spectroscopy in the far left, which responds fastest to changes in water availability. Then we have thermal data. After that, passive optical data, so the reflectance indices, which can actually determine if pigments change within a canopy. And then lastly, we have changes in the morphology, which can be detected by active systems. And what we wanted to do in this study is build a large data set to try to determine, build a large data set with high temporal resolution, so daily data, to try to determine if this can be detected earliest or earlier than thermal or passive optical data. Okay, so this connection we want to make. But as you heard before, solar-induced chlorophyll fluorescence is influenced by the canopy structure and by the illumination conditions. If we were just to take SIF as is, then we would, and if we weren't accounting for the illumination conditions and the canopy structure, we would not be able to actually track the physiological state of vegetation. And we do so, as Sebastian Wienicke already showed, there's a way of doing this using the vegetation indice NIRV. So the NIRV basically is our proxy for the canopy structure, and the illumination conditions are defined as par. And we combine this with SIF in the far red, so it's 740 nanometers, to derive the fluorescence quantum efficiency at leaf level, so phi f. This is then compared to subsurface water storage in our study, so a measure of soil moisture in the root zone. This is a variable that is modeled by also our colleagues at Research Center Jülich. It combines the common land model with the power flow model, and we get information about the water content in the soil at different layers, so between 2 centimeters down to 2 meters. And what we also get is anomalies. All right, so the motivation is fairly clear. The question is, can phi f be used to detect changes in this subsurface water storage anomaly? And we do this in Germany because the subsurface water storage anomaly data is available in Germany to us. And we did it for agricultural and for forest ecosystems. And then secondly, can we determine a lag between these two variables? We heard it already before in Uwe's introduction, that this lag between the reaction of the SIF variables to drought is very important. Okay, and the second step is basically, do other variables show similar temporal behavior to phi-f in detecting changes in subsurface water storage anomaly? So is there any merit in actually doing these calculations? Or do vegetation indices, so reflectance-based vegetation indices, and top of canopy SIF behave similarly? All right, the data and workflow, I go through this quickly, but we used in this study the TropoSIF product by Gonter et al. It's available at daily temporal resolution and at 3.5 by 5.5 kilometers. And here you already see that this is a very big difference to the talks we heard before. We're looking at ecosystem scale, basically. And this within the time frame from 2018 to 2023. Then this is combined with NERV, which is also calculated from Tropomi input data to keep with the same sun sensor geometry. And lastly, the third data set which we need is the PAR data, which we get from the Breathing Earth System simulator. We heard a talk by Ryu yesterday, but he developed this Breathing Earth System simulator where a radiative transfer model is used in combination with artificial neural networks to be trained on MODIS observations to then give us PAR. And this, again, is also available at the same temporal and spatial resolution. And then lastly, the comparison data set. And I want to highlight here again that we are looking at daily data or almost daily data, which is most important to us. The general workflow is fairly straightforward. We get the base data sets, and then we calculate the subproducts from it. And lastly, we do a resampling and masking to actually derive or get the pixels of all the agricultural areas and of the forests. And almost all of these processes are done in Google Earth Engine, so it's fairly straightforward to also change this to maybe some other areas which would be of interest. All right, so in general, calculating phi-f maps and then comparing them to subsurface water storage anomaly. And what we get is this, so a time series. Each data point in this plot shows the median of all available pixels within Germany, and we also applied a rolling average over 2 days to eliminate any cloud gaps that may occur. And in the top, you see now the subsurface water storage anomaly for all the agricultural and all the forest areas, the agricultural being blue and the forest being green. And here you can already see that in these 6 vegetation periods, we had a drought in Germany in 2018, and then consecutive drought years after that in 1920, then a wetter year in 2021, and then some drier years in 22 and 23. So this already reflects the truth. This is the rest of the data set now. So we have the 3 variables. So we have SIF up here, then NERV and PAR, so the input variables for the calculation of phi-f. And here you can already see the strong seasonality in all of these vegetation periods and the strong link between NERV and SIF, so top of canopy SIF. They behave very similarly in general. This strong seasonality is then obviously reduced once we normalize for it in the phi-f data. But the question now is, how do we compare the phi-f to the subsurface water storage anomaly to determine if it can be used to track drought? What we did is use 2 simple constraints. First, a negative anomaly that is longer than 30 days to determine specific timeframes within this data set. And in the agricultural areas, we then found 8 time periods, and in the forests, we found 6. These time periods were then cross-validated against the combined drought indicator by the European Commission. This is available at 10-day intervals, but we could already see the same pattern to be within these watch and warning periods for this drought indicator. All right, coming to my results now. We did a cross-correlation analysis to determine lag, so between subsurface water storage anomaly and phi-f for the agricultural areas and for the forests. This is what you get here. So if you were to take the time frames as is, or the subframes that we have, calculate the cross correlation, you would get the correlation at lag 0. And the rest of the lag is on the x-axis, which you see. So we see this pattern that emerges once we shift phi f into the future. We see that the cross correlation coefficient rises until a peak after 2 days, and then it drops again. In the bottom, the red dotted line you see is the 95% Monte Carlo envelope to determine if this could be related only to noise. But what we determined from this is that there is a connection between phi f and subsurface water storage anomaly with a lag of 2 days in the agriculture, but also in the forest pixels. Here we also see a very similar pattern, which we determined could be down to large amount of mixed pixels that we have in these 3.5 by 5.5 kilometers. So we assume that that is the case rather than this behaving completely differently. Now, the second question we had, is this also visible in the reflectance indices or in the top of canopy sieve? And this is what you see in these plots. So the phi f you saw before is the one down at the bottom, just with a different scaling. But if we focus now on the top row, so sieve, NIRV, and NDVI, we see very high standard deviation. We don't really see a strong pattern that emerges in the lag. So this goes down to 0, and then this is almost 0, at least within these 7 days, and in NDVI a similar thing. But the second thing we also did is we used LST from MODIS to cross-check if this is a variable that should behave at least similarly if there's drought conditions. And what we found is also this pattern that emerges with a rising cross-correlation after 1 to 2 days and then a drop again. But in general, an inverse relationship between LST, because LST goes up as the anomaly is going lower. And in the forests, we also see a very similar pattern. So the strong standard deviation within this 95% Monte Carlo envelopes for the top row, so for the reflectance-based indices and the top of canopy sieve, and then a similar reaction for LST as well. What this does not mean is that NDVI or NIRV or the reflectance-based indices and top of canopy sieve does not react to drought, but it doesn't react within these 7 days. If we were to extend this further, then we would assume that this would also react at some point. Concluding now, we determined that phi f can detect changes in subsurface water storage anomaly in this setting. So from the satellite perspective, we could determine a 2-day lag for agricultural and forest ecosystems. And we can determine that NIRV and NDVI on top of canopy sieve is not able to detect such changes. Then lastly, this again highlights, as we already heard in almost all the talks today, the importance of sieve downscaling or normalizing if we want to learn more about the actual physiological state of vegetation. And then lastly, what we also found is this, that LST responds similarly, or in some cases, even earlier than phi f to changes in subsurface water storage anomaly. This may be counterintuitive to the plot I showed in the beginning, but this is the theoretical background. And we do a lot of spatial aggregation and then also some temporal aggregation. So this not behaving exactly as we anticipated does not mean that we should exclude this. Now, my outlook looks as follows. We have created a European data set for phi f, which is available at this DOI and at the QR code you can see. Here we have included the bands we used for the calculation of phi f, but also all the qualities bands we could actually get from the TropoSieve product as well. And yeah, the connection to FLEX here is actually really important to us because it's very difficult to validate 0.05 degree resolution data. So having an intermediate satellite product that is the base product for sieve would be really important in connecting these findings that we have now found on the ecosystem level to actual ground data. Thank you for your attention.
Thank you, David, for this very inspiring talk. And we have time for a question. Yeah.
Hi, thank you very much for your talk. Just a question, you have two different ecosystems, very different,
Yes.
who probably are not reacting the same way towards rot. How do you explain that you're saying the same patterns? If you explain
we assume that that's down to a lot of mixed pixel effects that we have in this data. Because it's a 0.05 degree resolution, so 3 by 5 kilometers. So we assume that that's the case, that these fragmented forest pixels that we have in there also are not actually only forests, but also grasslands, or also some agricultural areas as well. So this is actually what we believe is the case.
Thank you.
Thank you very much, David. So our next speaker is, I'm sorry for the pronunciation, Steve Honke, sorry, from Forschung Centrum Jülich, who is going to present us her study on cyan-induced fluorescence and the effect of elevated CO2 and Cercospora leaf spot on photosynthesis. Thanks. Thank you.
Thank you very much. Yeah. Good morning, everybody. I am Dipti Koncha. I am a PhD student from Forschung Centrum under Uwe's group. And today I'm going to present my work, which is entitled as Sun-Induced Fluorescence-Derived Canopy Level Effects of Elevated CO2 and Cercospora leaf spot on Photosynthesis in Field-Grown Sugar Beet. So from previous talks from Uwe and others, we know that SIF has emerged as a promising tool for assessing the photosynthetic dynamics, but the use of SIF and for monitoring the biotic stress remains very underexplored. And in Germany, the sugar beet is a very commercial crop for the sugar production. And in this sugar beet, there is a fungal disease called Cercospora leaf. Sorry. There is a disease called, the fungal disease called Cercospora leaf spot, which can lead up to 50% yield reductions under disease severe infection. And here you can see how this disease looks. So these are circular reddish brown lesions with gray centers, and these lesions, they coalesce and they spread throughout the leaf and also throughout the plants, which finally leads to the whole death of the plant. So this disease, it affects the plant both structurally and physiologically. Structurally by compromising the light interception, and physiologically by damaging the photosynthetic performance. But beside its direct responses, the level of infection, it can also differ under different environmental conditions. So we also want to check how, in our experiment, we focus on elevated CO2 concentration, because we all know that in the future, next generations, the CO2 concentration in atmosphere is going to increase up to 1000 ppm in the next coming 50 to 75 years. So how we did this? So we had an experimental setup in Campus Klein-Altendorf, Bonn, Germany. And on the left side here, you can see that we have a system called FACE. It's a free air carbon dioxide enrichment facility where we can put a targeted concentration of CO2 up to 600 ppm. And beside this, we a bit far from this experiment, we also had an ambient setup where we did our experiments in ambient CO2 conditions, that is 400 ppm. And in these both setups, we have used two sugar beet varieties. One is susceptible variety, which means this variety gets infected very fast with Cercospora leaf spot. And the other one is very low susceptible variety, which means it is resistant or semi-resistant to the disease called Cercospora leaf spot. And under these setups, we divided each half of the setup. We sprayed it with fungicide, so we don't have a lot of disease or very low disease pressure. So from that, we have two treatments, that is fungicide under ambient conditions and fungicide under elevated conditions. And also the other half of the setups, we inoculated the disease. It basically means that we forcefully infected the plants to have high disease pressure. So from that, we get two more treatments that are inoculated under ambient conditions and inoculated under elevated CO2. So Next, so we want to see how this disease and yield, the final yield responded under this elevated CO2. So on the left side here we have this seasonal disease severity. The x-axis shows days after sowing, and y-axis shows the disease severity. And if you see these strong orange lines, in the susceptible variety, the disease severity went up to 8 by the end of the season. And if you see the green lines, which are sprayed with fungicide, the disease was up to 4. But when we look in the very low susceptible variety, as it is semi-resistant to the disease, the inoculated plots which are infected with disease, they were only up to 4 disease severity score. And in the fungicide plots, there was basically no disease or very less disease pressure. But also, if you observe the interesting thing, here the circle shows the ambient conditions and the rectangular, the elevated conditions. So the disease severity, they did not differ between the ambient and elevated conditions, which was also reflected in our SIF canopy measurements. And next, if we observe the final sugar yield at the end of the season, if you observe, here we have sugar yield on the y-axis and the inoculated and fungicide treatments under both ambient and elevated CO2 conditions. And you can clearly see that elevated CO2, it significantly increased the yield in fungicide-treated plots in both genotypes. But if you see under inoculation where there was a high disease pressure, the elevated CO2 didn't have a significant effect. So based on these two results, we thought that the elevated CO2 didn't have a strong effect on the disease. From now, in my next slides, I'll focus only on the susceptible variety under ambient conditions to differentiate the inoculated and fungicide treatments. So coming to my research objectives, first objective is to evaluate the detection of the CLS disease symptoms using passive sun-induced fluorescence. And in our previous work, we also did an experiment in Göttingen under biotic stress, but only under 2 disease infections. And from that experiment, we saw that the SIF response was strongly driven by structural parts than the physiological downregulation. And also, from previous talks from Uwe and Vinike, we also understood the complexity, the relationships between PS2 yield and the SIF yield, that under very high stress conditions, they are negatively related. So to understand both these concepts, my next objective is to disentangle this structural and physiological drivers of SIF responses, also in a combination with the active chlorophyll fluorescence using the seasonal data set. So how did I do these measurements? We used a automated positioning system called FieldSnake, which allows us to do high throughput phenotyping. And here you can have the close-up look of this. So we mounted both active and passive fluorescence sensors close to each other, and we did the measurements simultaneously, so we can compare the measurements from flocks and leaves together. And we used the fluorescence box from JB Hyperspectral to get the flux measurements, from which we used canopy SIF at O2A and O2B bands, and also PAR measurements and different vegetation indices. And we used an active sensor called light-induced chlorophyll fluorescent transient system, where we use this strong excitation pulse to close all the reaction centers, and from that we get the PS2 efficiency, that is FQFM. Then from the previous talks, we also know that it is very important to normalize this sun-induced fluorescence. So I got the SIF 760 and SIF 687 from the fluorescence box, but we calculated the SK fraction from the reflectance measurements of the flux box using the Juan Romero model from 2018, and we used this to divide the SIF with the APAR and the SK fraction to get the final fluorescence yield at 687 and 760, which basically indicates only the physiology, including the structural part. So coming to my results, so here we see some of the reflectant indices, that is FAPAR and NIRV on the y-axis, and days after sowing on the x-axis. And the first symptoms, it means that we saw these symptoms by our eye, and the symptoms were after 1 or 2 days after sowing. And you can clearly see that a fraction of APAR and NIRV, they were decreased in inoculated plots from early stages of infection. So it means that the vegetation indices or different parameters, they reacted to the disease very well. And now when we look at the canopy CIF values, so here you see that CIF 760, it is very proportional to the APAR, and it also decreased in the inoculated plots from very early stages of disease infection. And also when you look at CIF 687, the responses were similar in the later disease stages where the disease was prominent. But when you look at the early stages of the disease, you see that CIF 687 is inversely related to 760, which makes sense because of the chlorophyll reabsorption in the dense canopies. Then now we have, so this CIF, it indicates both the structural and physiology, and now let's look at the fluorescence yields, which is only the physiology part. So here we have the CIF 760 and 687 yield on the y-axis and days after sowing on the x-axis. And if you remember the plots before, there was a clear difference between inoculated and fungicide plots, but in the yields, you don't see that effect anymore, which indicates that this disease is driven strongly by structural effects compared to the physiology. But also the interesting part is in very later stages under high disease stress, both CIF yields, they increased in the inoculated plots, which indicates a physiological response. So now I have the active fluorescence measurements, and let's check how, when we compare it with the active fluorescence measurements, how does it look? So here I have PS2 efficiency on the y-axis and days after sowing in the x-axis. And you see that in very later stages, the PS2 efficiency decreased in inoculated plots compared to the fungicide plots, but which is opposite to the trend of CIF 760 and 687 yields, which also, which was similar in Van der Tol and Martini papers. But also the interesting part here is the CIF 687, which is red yield, it showed the stronger response compared to the CIF 760 yield. Yeah. So coming to conclusions. So reflectant indicants determining CIF, they showed a strong response to disease due to canopy degradation and declining photosynthetic performance. And CIF response was found to be driven more structural than physiological downregulation. And also CIF yields and photosystem to efficiency, they showed opposite trends under stress conditions, with CIF 687 yield showing stronger response compared to CIF 760 yield. And some of the takeaways are that these interactions between CIF yields, 760 and 687, they can be disease specific. So if you check them in different diseases, they can respond differently. So it is important to note that. And also the CIF responses, they should be further evaluated in diseases that primarily affect the physiology to compare it with the PS2 efficiency and to understand the relationships better. And also to check the diurnal measures, as Vinike mentioned before, to determine whether CIF is more responsive to short-term physiological regulation than to structural canopy changes. Thank you very much.
Thank you for a really nice, interesting presentation. There is any question?
Thank you for your presentation. I'm curious how you account for the canopy structure change, how to separate the canopy structure change and the physiology change. So how you measure the APA?
We got it from fluorescence box, flux box, the APAR measurements. Flux box. It also gives.
So you have the above canopy and the below canopy measurement?
No, I have only above canopy APAR measurements.
Yeah, I'm just curious. I doubt such kind of method could not fully account for the canopy change, canopy structure change, which could be mixed with the physiology information.
Yeah, that's what, so, yeah, here if you see FAPAR and NARV, they mostly indicate the structural path. But if you check the CIF, canopy CIF, it has both structural and physiological information, which also clearly showed the response to the disease. But when I down-regulated or normalized the CIF and checked the yields, we don't see that response anymore. So we think there is a physiological downregulation as well, but this disease, it affects the structure very prominently, so we don't see that. Or we may need very high-intensive time measurements to, or very low leaf level measurements to check this.
Okay, one more question is about how you calculate FPAR.
I have PAR, incoming PAR and the APAR from the FLOX, so I divided them to get the fraction of APAR.
Okay, I understand. Let's talk more later.
Yeah, thank you.
Okay, thanks, and thanks, Depti. I think it's clear that there are things we need to still explore further, because there is this inconsistency sometimes here and there, so somewhere there is room for improvement.
Thank you so much.
But yeah, thanks to all speakers from this session, and let's give an applause still. And then we can move to the coffee. Yeah.
So we have to watch out. People are not allowed to leave after our session.
Take it here. Exactly.
They normally know the microphone.
Okay. Thank you. Hello, everyone. Sorry, we will need to start soon.
Gracias.
Hi everyone. It's time to start our session on understanding the carbon and water cycles using fluorescence data. So I will invite you all to sit down and make some silence for our speaker. First speaker today, Adrian Moncholi Estronel, and he will present monitoring photosynthetic quantum yield through non-photochemical quenching from laboratory to field, integrating canopy fluorescence, reflectance, and GPP. The floor is yours.
Thank you very much for the introduction. First, I will try to cover all the previous work that we have developed until the next step will be to apply them to the field scale. I am part of the PhotoFlux project, which aim is to retrieve photosynthesis variables from the optical properties of the vegetation using a bottom-up approach that goes from the pigment absorption spectrum through leaf and canopy scale. We also are involved in the FLEX, which is developing sampling strategies for the FLEX mission, CalVal. Since the fluorescence and photosynthesis is not linear related, we propose that we need additional products that are necessary to interpret the fluorescence signal. And the MPQ, it's a key parameter related with the xanthophyll photoprotection mechanism, and it can be monitored with differences in reflectance. Previously, Van Wittenberg developed estimation of the xanthophyll-driven photoprotection absorption of the pigments with a model based on 3 Gaussians, which retrieve the effective absorbance of its pigment in order to go beyond the PRI estimation of the MPQ estimation through the PRI. This was adopted in the Pescador work, where we were able to see the in signs of photoprotective mechanism at leaf level in tomato plants, and those results were also validated with the pigment analysis at laboratory. We moved to the canopy level, where under control conditions in a growth chamber equipped with a fluorescence active measurement sensor and a spectral scanner, we measure reflectance and photosynthesis-related variables using the fluorescence active sensor. We monitor the quantum yield fluorescence, and we also were able to see differences when we adapt the plants to different light conditions during the diurnal cycles. Looking at the reflectance and absorbance, we can see really small differences. When we compute the difference accumulated during those phases, we get 2 main peaks, one in the PRI region and another one in the red edge. Going forward and applying the previous results, we put together a tomato dataset together with a Scotch Pines one, and where experiments were using similar instruments. Those instruments consist on also the veneer measurements and also fluorescence active fluorescence measurements. Since we retrieved these variables from the active camera, we apply the partial least square regression, which key advantage is that it provides an estimation of the importance of each wavelength for the modeling. That is, values that are higher than 1 means that those wavelengths are important for the modelization. Then we can see also the peaks that we were retrieving with reflectance deltas for both quantum yield and the MPQ. Also for both species, the tomato and Scots pine. So we can summarize that we can see differences in the PRI region and in the red edge consistently with reflectance and with these statistical methods that are able to monitor the plant photoprotection. And we have to highlight that these statistical models are very useful, or we propose it as a screening tool, but they are not resolving the underlying different pigment contributions to the spectral signature. So now we move to the field level and we ask ourselves, we will be able to reproduce this methodology at natural environment. So for that, we will use, we used the two datasets, one from the Tower of Majadas, where it is characterized to be a Quercus savanna, and this dataset was composed of 2 summer months, and another dataset from Manzanera Tower, which is a more dense forest with 1.5 years temporal time series. And the variables to retrieve from reflectance, or to go against the reflectance with the PLSR model, was the GPP estimation from the Flux Tower, and in case of Majadas, we also had the MPQ values from 4 leaves. And what we have seen during all these conferences, when we put together the flux measurements and the GPP values, values retrieved from the tower, they don't correlate very well. That, in fact, it's a very weak correlation. So we started investigating the spatial distribution of through the NDVI values, and here we can see a correlation between the average footprint value of NDVI from Sentinel-2 and its pixel NDVI, just to see if we were able to see any pattern or spatial pattern to say that we were not choosing the correct tree with the flux. And also we do the same correlation analysis, but with the flux NDVI, revealing that we have a really good correlation. So we start thinking about, it will be the BRDF effects affecting this correlation. So we apply a BRDF normalization based on the Ross-Lee-Magnan kernel model, and then after applying it, we can see that it was an improvement of the correlation, but it is still weak. But then we go for the hyperspectral point of view. This normalization will change the PLSR outcomes from the measurements. So now we will compare the long time series from Manzanera, where we get all the values we compute the PLSR for all the values of the dataset, and those normalized by BRDF, And also, as a safe check, we compute also the PLSR for the known values with and without the BRDF correction. And we can see that these patterns of the peaks that we were looking for were not changing too much. Also, we have to highlight that this model performs really low. So the performance was really low. And after applying it, it was improving a bit, but it was not a big improvement. Yeah, that's. Then we move to see if these patterns, or these long patterns, also was revealing something in the VIP score. So we get a comparison between the entire dataset and a short subset corresponding to a rain pulse where the plants were under drought stress, and then we get rain and a recovery phase. And first we have to see that the improvement of the models, because of this short scale, that the model is able to retrieve better the variable or to get better correlations. And we can see that there is a peak shift in the red edge, where here in the PRI it is broadening, but not the peak is there. Going for the comparison between Manzanera and Majadas, where both are two. Well, we can say that in short-term temporal series we get similar patterns in the VIP score, and also going from the GBB to the MPQ, we can see that there are also these peaks revealing the spectral covariation between for estimating the MPQ. But when applying the BRDF, we have a lower performance. To conclude, the PLSR methods reveals that we are sensitive to changes in MPQ and GPP at laboratory and field level. This tool we see as a very helpful screening tool for getting a clue on where we have to model these spectral features, and more future work it will be to apply the spectral mixing to correct from to estimate the effective absorbance of pigments at this scale level. And also the field sampling is something that we have to think about. Thank you very much.
Thank you very much for taking us a journey from the laboratory to the field. Are there questions? While you're thinking, I have one. In your laboratory study, you showed the two distinct peaks in your data because of the different light treatment. Did I understand this correctly, that this was really short-term changes of light? So what you see there was pretty rapid, within minutes or a few hours changes?
That was an estimation of having the plants adapted to the higher level of light and then measuring with the same conditions. So it is revealing how the pigments are adapting during, meanwhile.
Okay, so it was a longer, several days adaptation to highlight conditions.
Sorry. It was those diurnal scales along a drought stress. Okay. But in all the experiment, we can see how the pigments are evolving, changing the peaks to higher ones.
Okay, so this means the peaks which you see there are related to
two things:
yeah.
Short and long-term changes.
Okay, thank you. Further questions? Comments? No? If not, then you're here to the rest of the conference for further discussions. Thank you. And with this, I would say we move forward to our next presenter. Sorry for the name. Hui Su Feng from Seoul National University, and you want to go with us beyond photosystem 2, look on photosystem 1. I'm really excited. The floor is yours.
Thank you so much. Hi everyone, good morning. My name is Huai Zefeng from Seoul National University. My topic is how photosystem 1 dynamics regulate the electron transport in respiratory. The photosynthesis, we know have two main reactions. First is the electron transport, another is CO2 fixation. Even though it has a long history, but the electron transport in traditional models, for example, the FVCB model, was still empirical. Recently, SIF was widely used as the proxy for GBP via electron transport, but their relationship was oversimplified. It's kind of like the black box. And as previous presenter shows, the NPQ and PQ, which qualified by the QL, were found regulating the relationship between SIF and GPP, but the behind the principle was unclear. If we unbox the black box, you're gonna see it's so complicated. A lot of mechanism in a lot of processes in the electron transport, they interact with each other, and most of them are related to the PS1, but was ignored. Johnson-Berry model provides a mechanical and a simple solution for these processes in the electron transport. The key idea of this model is about the excitation balance between the PS1 and the PS2. It also provides a fluorescence module, not only for the SIF, but also for the active fluorescence simulation. So everybody come here for the FLEX satellite. You may wondering, can we apply this model to the ecosystem or the global scale? I also think this question, I found that there are 3 points we need to care about. First, this model needs biochemical info and a leaf or canopy temperature as input. You may say, oh, okay, it's not too much, but if you want to get these two informations, you need to solve the carbon and water coupling, you need to solve the stomata conductance, you need to solve the energy balance. So it's not that simple. And we need a radiation transfer model for SIF. And we need a full validation at the ecosystem for the JB model. It was seldomly done. So here come 2 straightforward questions. How to apply this model and what's the performance of this model from 2 perspectives, the fluxes and the hypothesis. Here is our set. It's located at the middle of the Korean peninsula. We have a tower in the respiratory. We have a meteorological measurement, we have eddy currents measurement, and we have a hyperspectral system for SIF measurement. Apart from the tower, I utilize the random sampling method together with the 2 PAM sensors to collect the ecosystem level PAM measurement. I call it because I conducted a stand in the respiratory. I collect 108 pairs one day randomly within the footprint for the sunlit and sunshade leaves. Okay, how to apply the JB model to the ecosystem scale? For our group, we developed this model for many years called the Breathing Earth System Simulator, BESS. We want to utilize the photophysical and the photochemical from JB model and the biochemical and the RTM part from the BESS. We combine them together, generate the BESS JB model. We call it PS1 explicit coupled process model. It's a part of the bigger version and a simulation platform for next generation satellite observation, including the FLEX. Okay, for the radiation transfer part of the BESS, we utilize the Beer-Lum's law for the simplicity and the speed because we want to apply to large scale. We separated the canopy into sunlit and sunshade. We considered the beam illumination and the diffuse illumination. Based on this framework, I developed a safe RTM as the equation shows here. And we know that I mainly focus on the 760 nanometers. We know for this wavelength is strongly influenced by the multiple scattering, but in the original BESS, we used the hard-coded constant, the effective scattering coefficient. It cannot well catch the multiple scattering. So I utilize the scope simulation. I used 750,000 simulations, developed a lookup table for this parameter. As you can see, the lookup table achieved R square at 0.95. It's pretty good. I'm satisfied. And I developed the Python version of scope. We will release it together with Professor Van der Tal later. Please stay tuned. And what's the performance of the GP model at the ecosystem scale? First of all, we want to know whether it is a good model or not. The answer is yes, definitely yes. First, we have no optimization for SIF and GPP simulation. We got 0.65 at 30 minutes R square, and we got 0.84 at the daily scale. And more satisfying is for the GPP. We got 0.9 at the 30-minute scale with no optimization with the default parameters. And comparing to the original FVCB, best 2 is under 0.5 at the 30-minute scale. So it's a good model, and it's worth further explanation, further investigation. So how about the hypothesis? I test the hypothesis with these following questions. As I showed, there are a lot of processes. I test them one by one. First, the key hypothesis, excitation balance between the PS2 and the PS1. Here is the PAM observation result. As you can see, the PS2 and the PS1, they're not one-to-one equal to each other, but they arrive at the quasi-steady state at a high illumination. We think this, and we didn't expect they perfectly equal to each other, but according to this, we can draw a conclusion that the PS1 excitation and the PS2 achieve some steady states. And for quantum yield part, as you can see from the PAM measurement, the phi PS2 decrease with the illumination, and the MPQ and the phi NO increase slowly with the illumination. But during the experiment, we found that this process is also should have PS1 involved through the state transition. That means the antenna transition between PS2 to PS1. If we turn on the state transition, we'll see, well, it looks similar to the PAM measurement from the model simulation. But if you turn off this setting, you can see the underestimation of the phi PS2 and overestimation of the phi PS1. So this means that the transfer of the energy from the PS2 to PS1 is influencing our conclusion of the quantum yield of the phi PS2. And I checked whether there is cyclic electron transport, the 4 I indicate in this diagram, whether there is indeed have this process. And according to the PAM measurement, I found there are no directly transition from, there's no transition point in this plot. And because we use the darker interval relaxation kinetics measurements, we have no absolute value. But we can see from the figure that it's almost linear, no transition. And according to the model result, we also found that for our set, we didn't find the evidence of the cyclic electron transport. And MPQ. Why we need to care about MPQ for PS1? First, I know there are some papers indeed try to connect the photosynthesis with the energy balance. And I did some experiment. I found it indeed cannot ignore it. But different from our understanding that most MPQ is happening at the PS1 instead of PS2. As you can see from this figure, the PS1 phi MPQ increased sharply at the early stage and achieve very high value at the high illumination. But the 5PS2 just slowly increased for our set. And the model also tells us the same thing, that the 5PS1 is the account for the most heat dissipation. And finally, we also output the safe signal. Here I show a seasonal variation of the safe signal from the PS1 and the PS2. As you can see, PS1 almost account for the nearly half, and at some time it account more than half signals for the total safe signal. That means we need to be careful when we're interpreting the seasonal pattern of the safe and the PAM measurement. Okay, here comes the conclusion. The BESS empowers the JB model simulation at the ecosystem scale, and the BESS JB model shows greater performance on ecosystem scale plus simulation. And mixed processes related to PS1 are playing important roles on understanding and modeling electron transport. First, we found excitation balance between PS1 and PS2 in our set. Second, state transition. The excited energy transfer from PS1 to PS2 to PS1 directly influences the quantum yield. And electron flow around PS1 was almost identical as the linear electron transport of PS2. There's no cyclic electron transport. That means maybe in other sets they have, but for our set there's no. And NPQ in PS1 activates earlier and much higher than PS2. Safe from PS1 account for more than half sometime. That's all. Thank you.
Thank you so much for this presentation. Any question in the audience?
Yeah, thank you for this interesting presentation, and I'm really happy to see that you are taking the PS1 very seriously, because I agree it's far from constant and we have to account for it.
Thank you.
I have many questions, so maybe we can discuss later, but just one now here. So you were measuring with the dual PAM, right?
I'm measuring with two PAM. First, the RS600, and another called multi-spec, developed by David Kramer.
Okay, okay. That's all. But the question was about the state transitions. So how do you simulate them? You were showing the effect of considering them or not considering them. So is this something you get from the instrument?
Yeah, it's a good question. We cannot directly measure it. We just show the simulation result to see it and compare with the quantum yield result. Because we found the quantum yield, we were very sensitive to this parameter. So we attribute the quantum yield, the uncertainty, to the parameter. We didn't have a direct measurement.
Okay.
Yeah.
But very important, yeah, because again, state transitions, but also PS1 contribution changes probably within leaves inside the plant over time, everything. So very important to look at that. Yeah,
yeah, thank you.
Thanks.
Any other questions? Okay, so we'll start. Thank you so much, Waite, for your presentation. We'll continue again with Seoul National University with Jinho Lee, and he will talk about the apparent CIF and GPP relationship at canopy scale. Thank you very much.
Hi, good morning, everyone. My name is Jong Ri from the Young Years Lab. I'm the PhD school student. So let me start. Our work. The title is "The shared APAR is rather than the physiological coupling. Explain the apparent CIF to GPP relationship at the canopy scale across the diverse ecosystem." Let me start the motivation. Yeah, this is one of the key study that examining the region, how the CIF and GPP are correlated. And then the message is very clear. The A part explains most of the variance between the CIF and GPP, and then once A part is removed, the correlation between correlation is decreased, and the yield correlation also very not that significant. So this suggests that the apparent CIF to GPP relationship may largely reflect the shared light absorption. Then even these kinds of research were conducted by the single site or single PFT, many studies, to use the CIF over the simple GPP proxy. We therefore need to determine whether CIF contain the individual independent physiological information. Or we know that, as Albert explained well, such a physiological information clearly exists at the lip scale. The fluorescence yield and the photochemical yield are physically linked through the energy partitioning. As shown in left, the coupling is well established by the photosynthesis theories. And the key question is whether this relationship is preserved when the scaling from the lip to canopy scale. But however, we examine the canopy scale observations, the pictures become less consistent. A study during the 2018, the European heat wave suggests that the lip scale measurement, lip level results are the same with the canopy scale result. But another study like ABA experiment showed that the stomata-derived stress reduced the photosynthesis, but the CIF remained largely unchanged. This finding indicates that the VF and VP relationship at the canopy scale is not straightforward. And this means that something changed when lip level process are scaled up to canopy scale. The previous literature suggests that the canopy structure may explain this difference. The canopy is not a single leaf. It is contained both sunlit and shade leaves under the very different light conditions because this vertical light gradient and the energy partitioning changed within the canopy. And when all the leaves are combined, the canopy level VF response can look very different from the all individual leaves. In short, the canopy structure could change how physiological signal appearance at the large scale or the canopy scale. So, however, additionally, the canopy structure is not the only factor that can change this H yield. So environmental stress can also create the divergence between the VF and VP, and stomata close may create the asymmetric between the asymmetries between the VF and VP. Whether this pattern is consistent or not remain as unknown. So address this uncertainty, define the gap of our study. So in this study, we will address these three questions. The first is, does shared APA absorption explain the apparent CIF to GPP correlation across the ecosystem? Because that kinds of oral research are conducted at a single site or a single PFT. And then our second question is, does canopy light region reshape the apparent light response of the VF at the canopy scale? Our third question is, how does canopy scale VP and VF response differ to the environmental stress across the ecosystem? Let's move to the methodological part. We measured the CIF at the 5 different ecosystems at the Korea, the cropland and the wetland and deciduous forest and needle forest. Additionally, we measured the mixed forest too. The observation period are ranged from the 1 to 3 years, usually 2 years or 3 years at the growing season, and depends on the site. Depends on the site. And then the site characters are summarized on the right panel. And as we measured the canopy CIF and using the water prism system, as Young-Yeol mentioned yesterday, we used the bi-hemispherical observation to the accounting for the spatial heterogeneity and the temporal shift in the GPP flux footprint. Then our framework for investigating the SIF to GPP relationship, we use this framework for the decomposed both SIF and GPP at the three part. SIF is expressed as apocolopil and the VF and the escape ratio, as we all know well, and then GPP also expressed as apocolopil and the VP and the EUE, the energy use efficiency, and the equations are following the right. Then we examine how SIF and GPP responds to environmental drivers. One important variable is the KT, the clearness index. It is represent the how the sky is. It is calculated by a ratio of incoming short-wave radiation to the extraterrestrial radiation. The high KT, high clearness index, indicated that the dominance of direct radiation means that the activation of the sunlit, and low KT indicate the diffuse dominant conditions, indicating that the contribution of the shade lit. So therefore, we use the KT as an indicator of the balance between the sunlit and shade lit within the canopy. And then the SIF is retrieved using the IFRD for considering the atmospheric conditions. And then the GPP was calculated by nighttime temperature partitioning method to ensure the robust PM, the PM inversion for the canopy conductance calculation and the stable, the escape ratio, the estimation, and the overall the data quality. We limited the analysis to 9 a.m. to 3 p.m. solar time during the peak growing seasons. For achieve that, we develop one new model for the estimating the apocolopil. So we estimate the apocolopil using the neural network trained on the scope simulation. We estimate the apocolopil converting factor rather than the apocolopil itself. The reason is some ML or DL-based models, machine learning or deep learning-based model request some pixel-by-pixel calibrations. But in our model's case, 1 minus per albedo have the data lower for the baseline. So this kinds of model don't need to calibrate the pixel by pixel. And the input was the vegetation indexes measured by hemispherical reflectance. So NDVI and AVI represent for the greenness or canopy structure, and NRV for leaf angle, and the CCI and FCVI is the chlorophyll contents, and the KT is the clearance index for the atmospheric conditions. And all inputs are calculated by these bands, and then as the results show, looks nice at the scope simulations. The WD RVI and the EVI is used well for the apocolopil, but our model's performance is better than that, so we use that. Additionally, we first used the correlation and the partial correlation analysis. Then additionally, we examine how each yield responds to environmental driver. We apply the generalized additive method. This is necessary. That's because the PAR and temperature and VPD and the clearance index are very strongly correlated under the natural conditions. So a simple regulation cannot separate this individual effect, but the GAMs allow us to estimate the partial effect of each drivers additionally, to further reduce the confounding effect, we limited the condition like a temperature as a 20 degree to 30 degree or VPD to 0.5 to 2. And then for the environment stress conditions analysis, we just used energy saturated conditions and the clear sky conditions. Yeah, let me show the result. The first, we examined the SIF to GPP relationship across the 5 ecosystems, or the sites showing the positive correlation, but the slope differ among the ecosystem. This indicates that the single linear model cannot describe the relationship universally. To use the SIF as an independent potentialistic variable rather than the proxy of GPP, we decompose the SIP to GPP relationship into the structure part or physiological part. And then, across the canopy, across the ecosystem difference, the SIP is largely driven by the structure terms like the escape ratios, but the fluorescence yield remain stable. In contrast, the difference in the GPP are mainly caused by driven by the physiological variables, because these cross ecosystem driver differ. The observed SIP to GPP correlation is unlikely to arise from the VF and VP coupling. We tested this directly at the seasonal scale. The SIP and GPP seasonal scale and SIP to GPP are strongly correlated. However, when we control the APAR, the correlation dropped to near zero or non-significant or reversed. The APAR is strongly correlated with both SIP and APAR, but VF and VP correlation is weakened or non-significant. And at the midday scale, the pattern become clear. The SIP to GPP correlation weakness and then APAR to GPP relationship decrease more stronger than the SIP. As a result, the VF and VP showing the negative correlation at the most site. But according to the energy partitioning theory, the midday data should be into the MPQ phase. The remote sensing can observe. But our results, VF and VP, are expected to increase or decrease together. However, our observations results showing that the negative relationship between that yield, even under the non-stressed condition. This means that the process at the canopy scale changed the behavior we expect from the leaf. To understand what happened in the canopy scale divergence, we first look at the PAR effect on each yield using the GAMs. For the VP case, the pattern is same with what we expect, is decrease following the increasing PAR. For the VF's case, however, the response differ among the ecosystem. Some sites show the decrease or non-linear pattern, even non-stress condition like a temperature and VPD range. Even when we focus on the midday range, this divergence become clear. Under the high light conditions, like actually the 1,500 micromole is not that too much, not that too high. But even as that, VF is increase and show little decrease trends with the PAR. This behavior is consistent with the negative, our result at the result 2. To separating the pure effect of PAR from the canopy light region, we include the clearance index in the model. And as I mentioned before, the KT, the clearance index, represents the canopy light heterogeneity by the balance between the sunlit and shade leads. For VP's case, the overall decrease pattern is remained, but the magnitude is decreased. It was the same with what we expect. But however, the VF case, the normality, the behavior is disappear, and all sides showing the consistently the decrease with the increasing PAR. This shows that the apparent PAR response of the VF are strongly reshaped by the canopy light heterogeneity. What we found is that after accounting for this is what we found that. Finally, we tested how VP and the VF responds to the environmental stress, but the result actually is not that good because the environmental conditions are very different across the site, and we just used the 2 or 3 years data, and we focus on the narrow peak seasons range and the condition also too. So even so, the Phi-F variation are much smaller than Phi-P, typically within about 10 to 20%. So this is summary and take-home message. So the first, the across 5 ecosystem, that the apparent SIF to GPP relationship is primarily driven by the APAR observation rather than VP and Phi-F coupling. Second is canopy light heterogeneity reshape the apparent light response of the Phi-F, but once structural effects are accounted for, the Phi-F reveal the physiological signal by the PAR than the stomata stressed. Finally, although the Phi-F show the weakened sensitivity to the environmental stress than FEP, it retained the physiological information without the canopy structure effect, supporting using the SIF as an independent potential variable beyond the simple GPP proxy.
Hey, thank you very much.
Thank you.
Sake of time, is there an urgent question to the talk? If not to compromise with the lunch break, I would say you're around for more discussions during the lunch break later on. And I would like to move on to the next presentation, which comes from a group of the University of Paris by Zhao Yu Li. The floor is yours. Now?
Sorry, just to let you know, there will be a group photo after. I just wanted to make sure that everyone is not leaving the room at the end of the session. So let's announce it now, so you're staying here for the group photo.
Understood, so nobody goes out of the room before we have a group photo. Good. Now, the floor is yours.
Well, thank you. Hello, everyone. My name is Zhao Hui, and today I'm very honored to present our work, atmospheric dryness effects on canopy fluorescence and GPP in deciduous forest during the heat waves. So fluorescence is an optical signal directly linked to photosynthesis, and previous studies has reported its strong correlation with GPP across multiple scales, and we can measure the chlorophyll fluorescence at 3 levels: leaf level, canopy level, and global levels. The canopy level is especially useful because we can measure we can have the high temporal resolution fluorescence continuously, and it can connect the leaf level and the global level measurements. And then last, we can use both passive and active measurements method to get around some of the limitations of using just one method. And in this study, we use both passive and active method to measure the chlorophyll fluorescence at the canopy level. So one method is to capture the sun-induced chlorophyll fluorescence SIF, and another is the laser-induced chlorophyll fluorescence leaf. Similarly to the light use efficiency model, SIF can be expressed by the product of the PAR, FA PAR, and FU leaf, FY and F escape. The leaf system can focus more directly on the FY, and so we call that FU leaf. FU leaf can give us fast and detailed data, and isn't affected by the reflected sunlight, which is very important when we are trying to understand how the plants respond to stressful weather. So here comes our questions. How do SIF and FU leaf vary under different atmospheric dryness conditions? And how SIF and FU leaf are linked to GPP under high atmospheric dryness conditions? We did our measurement in the Fontainebleau bubble forest research station. This station is in the south of France, like 50 kilometers away. And this station is part of the ICOS network in France. And at the top of the tower, we have an eddy covalence system to capture the carbon flux, and passive sensors to measure the SIF, and active sensor to measure the FU leaf. We picked the two kinds of the dry air conditions, high atmospheric dryness had and no atmospheric dryness net. Consecutive days of high VPD were selected as the had conditions, and the 2 consecutive days after the had and the without rainfall were selected as the net condition. Before go further, we analyze the difference of the environmental factors and the phenology stability. And this figure shows that there's a significant difference in the environmental conditions like light and temperature, air temperature and the VPD, while the soil water content levels are similar between the head and the net conditions. And so we think that the soil moisture remain stable across both periods, and it was unlikely to be a limiting factor of our study. And as for the canopy structure, we checked the canopy structure, the violations of the canopy structure, chlorophyll content, and the light absorption capacity. CV is the coefficient of violations, which is very small at both half-hourly and daily scales. So we think that the plants didn't undergo substantial phenology changes during our study period. Here are some results. So under hand condition, GPP peaked earlier than both the PAR and the SIF peaked. It indicates that it's a stress-induced decoupling of the photosynthesis from the light availability. However, the FU leaf dropped a lot during the dry days, reflecting the down-regulation of the efficiency. But it remained quite stable during the net conditions. We compared the 4 correlations at both at the half-hourly scales. We can see the strong impacts of the atmospheric dryness stress on these correlations. But at the half-hourly scale, this data looks quite noisy due to the influence of the dino violations of PAR. Let's look at the daily scale. At the daily scale, we can see the stronger correlations of FU leaf versus SIF and GPP versus FU leaf under the head condition than net conditions. And stronger correlations of GPP versus FU leaf than GPP versus SIF under the head conditions. So FU leaf showed the strongest correlations with photosynthesis under the head conditions. To dig deeper, we use the light response curve method to calculate the Amax, which is the maximum rate of the photosynthesis. This method can separate the light from other environmental factors. From this figure, we can see there are stronger correlations between the Amax versus the FU leaf than the Amax versus the SIF-Y. And the strongest correlation is the Amax versus SIF under the clear sky conditions. So it shows that it shows the advantages of FU leaf in capture the plant's photosynthetic response to abiotic constraints. At the end of my presentation, here are two take-home messages. And first, the correlations between SIF and GPP weakens under the increasing atmospheric dryness, while the relationship between FU leaf and the GPP strengthens. Second, FU leaf outperforms passive chlorophyll yield in checking photosynthetic capacity, Amax, especially under the conditions of high atmospheric dryness. Our future's work will focus on having more measurements at other ecosystems and simulating the photosynthesis inhibition by abiotic factors using models. Thanks to our courses and my founders from Cannes and Sealand. Thank you very much.
Thank you very much. Have any question about this method that I really like, actually?
Thank you.
Question in the audience?
I really enjoyed your talk. This is a super cool technique. And I was curious if it has been possible yet to reconcile the photochemical yields from the lift technique with photochemical yields from PAM measurements.
Sorry, can you repeat your question?
You were emphasizing the potential of the fluorescence yield derived from the active measurement. And one of the things I've always wondered about is the comparability of the equivalent of photochemical yield determined by the active technique, and whether it's really possible so far to relate that quantitatively to a photochemical yield from PAM measurements.
I think Jane's question is about can we do the same thing at the canopy scale that we're doing at the leaf scale, and getting towards this kind of like quantum yield for PS2 and so on, just using your technique on canopy scale. Did I get it correct?
Yes, the answer is yes, but for the PAM, you cannot measure it for a long time, continuously. So if you want to analyze the seasonal violations of the leaf of the fluorescence yield, you you need to measure it at the canopy level.
Why don't I will follow up with you afterwards to try to articulate the question a little more clearly.
Okay,
Thank you so much for your talk.
Okay.
Is there any other questions in the audience? No. So, Joachim, thank you very much for your talk. And now it's my pleasure to introduce Aaron Liu, who is at Imperial College of London, working with me. So, really proud to introduce him and his work on ECMIP2 phase 2. ECMIP2, I'm saying, I'm just confusing. SIPMIP and the phase 2 of SIPMIP.
Thanks, Claire. Thanks for this brief introduction. And this is Horan. And today my talk is to introduce the current progress of the SIF model intercomparison project progress. It basically is the second phase of this unfounded project. So this project is co-led by myself and Nick, and we have collaborated with different model teams and observation teams from different institutes across a global scale, and we really appreciate their contribution to this SIFME project. So the motivation of SIFME is basically because SIF has big potential to track GPP dynamics. However, its relationship is dynamic and it is not consistent and static across different environment conditions, temporal scales, and biomes. So, like this figure showed, the SIF and GPP relationship tend to be nonlinear at our scale, but when scaling up to daily or monthly scale, it could be more linear. So we need to thoroughly understand how this dynamics could be, and so that we can better utilize the SIF data to better understand, track GPP dynamics and better understand carbon dynamics within the terrestrial ecosystem. So, however, the current SIF measurements are limited by either spatial and temporal scales, so that we have to find another alternative way to have more SIF data to improve our understanding. An alternative way is to use model-based SIF simulations. However, like this figure 1 showed, the SIF simulations also exist large uncertainties across different model configurations. This is motivate us to do a SIFME project to better evaluate and to see whether if we can firstly extract the uncertainties in SIF simulations so that we can finally improve the water and carbon flux exchange and how improve our understanding of them within the terrestrial ecosystem. So SIFME basically we provide the modelers to the model inputs like the meteorological forcing data, as well as some key vegetation information like the structural information of plant functional traits, etc. For the modelers information. And then we expect to collect some key predictions like the GPP, SIF, APAR, etc. Then we compare them with tower-based measurements. So for the phase 2 status, we select 3 air-grown needle leaf forests as a study area, and we also collect several model simulation runs at this size. And speaking to the SIF simulations within the model, I would like to give you an example based on the text model developed by myself. So here is the figure for the schematic overview of text model, and there are 3 key components which are really correlated with the SIF simulations. The first one, we rely on the photosynthesis to simulate the fluorescence efficiency, and then we scale it up to the canopy sieve emission using the both leaf and canopy radiation transfer processes. And also, the output is not only limited by the sieve. As I mentioned before, it also contains other key ecological variables like GPP, APAR, and photosystem level quantum yield, light use efficiency, and sieve yield. So here is the results of model comparison benchmarked by the tower-based measurements in here. So these colorful figures represent the statistical indicators, which is the biases between the simulations and observations. Here I just want to give you an intuitive impression of how large difference among them. So it doesn't really matter to remember or keep in mind these figures in here. So when we focus on the sieve part, we can see there is a large inter-model variability of diurnal patterns of sieve, with some models like the OKD model overestimated, and others like the JOICE at narrow reach sites underestimated. We also plot statistical metrics for R-square value and RMS value. And from these two figures, we can see the results shows the GPP simulations perform slightly better than sieve. It means our model have a more better performance in simulating GPP instead of sieve, which suggests we may need more work to improve the model structure and the parameterizations of sieve parts. And also, we observed the site-dependent difference of model performance, like the sieve simulations at narrow reach do not perform as well as the others. So now we know the sieve simulations show distinct diurnal cycles, and we also know, okay, the model simulations differ substantially in absolute magnitude and variability. The next step we're thinking about is how could we improve our model performance, and how could we reduce this model performance uncertainties? The two experiments to answer this question are designed to exercise how, if we could utilize the data, I mean outside data such as satellite data, tower data, to better help us to improve our model performance. For the first example, we use OKD model with and without model parameter, optimize the model parameters to see whether there is differences with each other. And for the second experiment, we use the BEPS model to optimize its parameters using the tower-based sieve measurements in here. Here is the two models experiments results, and for the figure 1, it showed how the OKD model performance before and after model optimization, and for the second figure, it showed how the best model performance before and after model parameter optimization. And all these are evaluated based on the tower-based measurements. So the general conclusion from here is after the data assimilation, both sieve and GPP substantially reduce the biases. So it brings simulate diurnal GPP and sieve close to the tower-based observations in here. Okay, so overall, the second phase of sieve MIP offer a systematic assessment of sieve simulations against the continuous tower-based observations as this 3-algal needle leaf forest. And we also found, okay, there is a large differences in sieve simulations, which reflects our structure and parameterization issues. And there is need a lot of more work to continually improve both of them from both ecosystem level, leaf level, and canopy scales. And finally, our findings underscores urgent to leverage outside data like satellite safe data to constrain our model and optimize our model performance, which will be helpful for us to better utilize the safe data to the future carbon projection under the climate background. Yeah, that's it. Thank you.
Thank you very much, also for staying well in time. So this means we have time for questions. Sure.
Wondering, can you tell more or less how many different SIF models you had in, I mean, by SIF model is SIF, ways of modeling SIF in these different models. So you had the Johnson and Berry, you have Van der Toll, any other ways of modeling?
Yeah, that's a good question. So actually, the difference can be classified into several parts. The first part is based on the photosynthesis part, and some model like, most of model using the FACA model and combines the fluorescence modeling to calculate the fluorescence emission. And however, some model like my model adopt Jen's new updated photosynthesis model to calculate the efficiency. And secondly, it really depends on what really transfer concept you adopt in your model. And some model use the widely used scopes for stream retransfer model, attributes like the leaf and the canopy scales. And for me, I use a spectral invariant theory-based retransfer model. So all of this structure difference will result in your SIF simulations be different to each other. Yeah.
Okay. Still no questions from the audience. I would have one, but that's really an understanding question. Did I understand it correctly that your intercomparison between the different models were done on different data sets? So each model used the data set where it was parameterized? Or did you use one reference data set where you applied all the models?
Basically, yes, we provide uniform forcing data to our models.
Okay.
Yeah, but for the parameterizations, we have provided our modelers some reference, and they can decide it, to adopt or not based on their own model situations.
Okay. Okay, this was it. Thank you. Thanks for this clarification. Good. Thank you very much. Then I think we move on. Thank you. And the next presentation is given by the University of Valencia, Pablo Reis Munoz. And you are, again, going, if I understand this correctly, on the larger scale up to tropomi-derived SIF and Sentinel-3 data. Please.
Thanks. Yeah, so, yes, I'm going to present the work we are carrying out using global troposif data along with Sentinel-3 vegetation products for prediction of GPP and the interrelationship of these variables that we found. Okay, so SIF is the most direct measurable indicator of the photosynthesis activity from satellite linked with the partition pathways of the absorbed solar energy by plants. At canopy level, SIF and GPP are positively coupled, given that they share a common driver, which is APAR, the absorbed photosynthetically active radiation. Yeah, this coupling can break up under stress conditions. APAR depends on canopy structural variables such as LAI, FAPAR, and also meteorological inputs as the photosynthetically active radiation. Then LAI and FAPAR can be derived from Sentinel-3, which will be flying in tandem with FLEX, so this information will be crucial for the interpretation of the SIF signal and GPP. This schema illustrates the interrelationship of the variables we want to study. So we have meteorological inputs that influenciate vegetation dynamics, which in turn produce changes in SIF and GPP. So this is SIF and GPP is indirectly controlled by meteorology, while SIF is a proxy, is correlated with GPP, so we can use it as a predictor future. So with this, we aim here to advance in the interpretation of the link between SIF, GPP, and meteorological constraints through satellite-derived products. In this line, we aim also to intercompare different GPP products driven by diverse inputs, either using satellite SIF or either using soil meteorological information. And also, in this line, we also aim to analyze causality or correlation between diverse meteorological constraints on satellite SIF response at large scale. To study the interrelationship of these variables, we have different statistics. We have Pearson correlation, which measures linear covariation. We have Granger causality, which introduce lagged effects and directionality. We can study bivariate analysis, so one variable causing response in the other variable, and multivariate, which can be applied to a combined set of inputs that cause response in the target. And we will apply also statistical significance through multiple tests by also assessing p-value and hypothesis test. So this is the data we are using in this study. We have troposif, which is daily representative, 7 and 3.5 kilometers spatial resolution with global coverage. Then we have meteorological information coming from the ERA5 land reanalysis product at 9 kilometers spatial resolution with global coverage. And then we have two house-made GPP products, which relies on the same algorithm, but which is trained with different data sets. We have the hybrid scope GPR GPP model, which is trained through scope simulations. Scope can simulate SIF and GPP, so we train our models. It is driven by troposif, Sentinel-3 derived vegetation traits, and ERA5 land product. And then we have an empirical approach through the eddy covariance GPR GPP model, which use field data at eddy covariance flux towers in order to train the models. This model is driven by a wider set of meteorological information from ERA5 land and also by MODIS derived LAI. The method behind is Gaussian process regression, a machine learning method that allows studying the band ranking analysis and that provides epistemic uncertainty estimates. When we train a Gaussian process regression model, we obtain a set of length scale, band-depending weight coefficients that can be used to study the relevance of the bands or the predictor inputs in the model. Once we train our models, we can apply to real data streams. Here we use the Sentinel-3, Sentinel-5P products along with meteorological data, and we implemented our models in Google Earth Engine for large-scale processing. And what we obtain is the model estimates and the model uncertainties that allows to assess the reliability of the model by pixel. We produce global time series for the year 2019 when using the scope GPR GPP model, and then 20 years of global time series when using the eddy covariance GPR GPP, which is driven by MODIS, LAI, and ERA5 land. This is a picture of the scope GPR GPP model. We observe the polar plot, which reflects the relevance of the inputs for predicting the output variance of the model. We have LAI, SIF, FAPAR as higher relevant variables. And then when applied to the real data stream, we obtain these maps. This is the yearly GPP values, which follows the expected spatial distribution along with its uncertainties. We here applied a test with different model configurations, testing different input configurations at the maximum spatial resolution of 300 meters. We tested the reference model with the 8 variables shown before in the polar plot, and then we remove SIF to see the impact that this causes in the model reliability. And we see that the uncertainties over the Iberian Peninsula increase. So this suggests that SIF, troposIF data is giving consistency to the model. And this is a picture of the eddy covariance GPR GPP model, which, as said, include a wider set of meteorological inputs, including soil temperature, soil water content, latent heat flux, precipitation, vapor pressure deficit. And then again, we obtain also the consistent spatial distribution along with its uncertainties. In this case, we have higher uncertainties on the northern latitudes that may be related with snow issues impacting the LAI MODIS product. And so the final goal was to intercompare these two different modeling approaches through this Pearson correlation map and the root mean square error. We see that for those areas where we have good match, this suggests that troposIF, along with Sentinel-3 derived vegetation products, may act as proxies of meteorological variables for predicting GPP. We analyzed more in detail the intercorrelation of these predictor inputs. We analyzed ERA5 combined meteorological information against SIF, LAI, FAPAR, also from SIF correlation against GPP, LAI against GPP, and FAPAR. And overall, we had a good correlation in general, while in some areas, for instance, in the here in the tropical regions, we have a decoupling of SIF and GPP, probably influenced by the canopy complexity. Okay, so finally, we also applied groundwork causality of a combined set of meteorological information explaining changes in SIF. We applied two different configurations, only ERA5 land data and also combined with SIF. We explored the statistical significance and the F statistic, and we observed that when adding LAI, the significance of the causality drastically increased. And then finally, this plot shows the spatial frequency of the significant causality distributed by land cover types, observing that in the deciduous forest and open forest, we had higher significant causality. Okay, so finally, the outlook of this work. We work in the interpretation of the SIF dynamics from satellite data, particularly as a predictor of GPP. we obtain a good match between two different GPP modeling approaches, suggesting that TropoSIF and Sentinel-3 derived vegetation products may proxy meteorological information for prediction of GPP. TropoSIF data enhances the consistency of the hybrid GPP models, reducing the epistemic uncertainty. Granger causality helps to identify sites where satellite shift dynamics are caused by combined meteorological dynamics. And finally, this work was conducted experimentally for 1 year of TropoSIF data. More work is expected when FLEX data will be available. So thanks for your attention.
Thank you, Pablo. Any questions for Pablo?
Yeah, thanks for your talk, Pablo. I was wondering, you said that SIF is important to predict GPP, and then you excluded SIF, but we saw in many presentations before that SIF is mainly driven by the canopy structure and by the illumination conditions. And I was wondering if you downscale your SIF product, if this FQE, or however we want to call it, the SIF yield, is still important for your prediction. Did you test that, or not?
This is a good question. We still didn't go into this detail. What we did, one of my colleagues, is downscale the spatial resolution of the TropoMi data at 300 meters, but we still didn't explore other detailed variables relating with SIF. So here we were using gross SIF data without normalization, but it will be an interesting future work.
Okay, thank you.
So
Thanks. Thanks, Pablo. Nice talk. I was wondering if you could tell us a bit more about what you said at the end with the grandeur causality, that when you add LAI on the ERA5 land, that you have more significant pixels. Can you tell us a bit more on what's your take on that? I mean, because there's not many pixels that are grandeur causal when there is only SIF. So a bit in the same line, maybe, that what Bastian was asking is, are we, I mean, yeah, in these maps. So if you're adding LAI, what exactly should we read out of this? That the LAI is helping, saying where SIF is useful?
So at the end, this is what drives SIF dynamics. When relying only on meteorological information as a predictor of the SIF changes, then not, well, a substantial portion of the area is significant, but this increase much more when adding LAI in combination with meteorological information. So the meaning is what are the drivers causing significantly from the statistical point of view the SIF dynamics.
So then I guess maybe the thing there, understanding that, is that since ERA5 doesn't have any LAI dynamics, it's a prescribed LAI. Basically, yeah, you're doing this, you're actually giving this phenology, because the LAI within ERA5 land is it doesn't change from year to year. It's prescribed. So actually, yeah, then it makes sense. Yeah.
Yeah. Mm-hmm.
Okay, right. Thanks.
That's right.
Okay, so we will thank you very much, Pablo. We will move on to our next speaker, Ingo Ensminger, and he will talk about tower-based shift monitoring of drought stress in Scott pines photosynthesis.
I can see the presentation here, but it's not on the... Which one? I'm already at slide number 4. Now it's gone. Okay. That's a different title than I thought, but anyways, my name is Ingo Ensminger. I will talk about disentangling the relationship between solar-induced fluorescence and photosynthesis in cod spine in response to drought. And contrary to so I will talk about a site in Switzerland. I'm actually based in Toronto. And the reason that I spent a little bit of time working in Switzerland is that I've been working with Arthur Gessler from the WSL in Switzerland for quite a few years. And they run a really, really cool experimental field site, the Fienwald experiment, and I will introduce that in a few seconds. And it's the scale of the work they did was very different from what you heard previously on the previous slides. It's nothing about global GPP or anything like that. We are just interested in a very simple question: can we distinguish drought stress from non-drought stressed cod spine trees in this experimental field site? So as I said, I will give a little bit of background on the Fienwald and work that we did there before. And then I will talk about the newest kind of experiment that we tried to do there with the solar-induced fluorescence measurements using FLEX boxes. And then I wrap up very quickly. So this is the Fienwald field site. This is an aerial view from the experimental plots. There are multiple treatments. This is the Fienwald itself, and in a very beautiful landscape. It's in the upper Rhone Valley, and behind that are the Alps. Inside the Fienwald, there are multiple observation towers, and on the figure right here, you can see these white little squares. Each one of them is a tower that actually gives researchers access to the canopy. This is Arthur Gessler demonstrating some stuff in the forest, Markus Schaub and Leonie Schöneck getting ready for some gas exchange measurements, and Petra Dodorico with a drone with a multi-spectral sensor that we used a few years ago in order to distinguish drought stress from non-drought stressed trees in this forest. So what you see is actually three treatments. There's an irrigation treatment in blue, a stop irrigation treatment. This is where irrigation was done for 10 years and then they stopped with the irrigation. And then in orange, there are the control plots that are non-irrigated plots. And this entire forest is actually the location where all the trees are naturally drought stressed. So in contrast to most of the other experiments where you try to prevent rainfall and stuff like that, here you deal with an environment where you're naturally limited in water. And all these trees, they're roughly 100 years old and they're barely 12, 13 meters tall. It's a really, really cool environment. So we flew this drone over the site, and this is a multi-spectral image from August 12th, 2020, over this forest. And what you can see quite nicely, all these dark colors, all these bluish tones, this means these are trees or canopies that are not drought stressed, where the PRI is rather high. And then in this control plot, you see there comes this reddish color, this reddish-orange color into the plots. And when we average the pixels from each tree, then we can nicely see how all this unfolds. And this was really, really cool. When we did the experiment, we thought, oh, this is something where we can use the PRI in order to distinguish drought stress from non-drought stressed trees. And when you average all those numbers, you can quite nicely see that the PRI works in a very nice way to distinguish all those trees. There was supposed to be another slide that is also gone. In any case, so that's the Fienwald. This worked out quite nice. Then I ran into Uwe Rascher a while ago. And then we thought we should probably install some FLEX boxes, which we did. So we installed those in order to figure out if we can actually distinguish stress-induced changes in the SIF signal from phenological changes in the vegetation. And we used an approach, or currently using an approach, all this is more or less preliminary stuff, where we use leaf level physiology, lichen measurements, leaf spectral reflectance measurements, photosynthetic pigments, leaf water potential, a range of parameters that are derived at the tree level, including subflow. And then there are canopy and above canopy measurements like tower-based SIFs using two fluxes. drone-based multispectral measurements. There's one guy who flies every 4 weeks over the forest and maps out everything quite nicely. And then in August, we had 2 high-plan flights over the site. And there's a poster from Zuniva McKeever and Bastian Siegmann in the back. I don't know the number right now, but I'm pretty sure you will find that actually deals with these high-plan data. And then there are lots and lots of environmental and soil data. So this field site looks like an emergency room. There are cables everywhere. And I'm only using a small subset of those today. This is a little summary of what happened in 2024 when we tried to set up these FLEX boxes. We arrived on June 8, and then we spent a couple of days trying to set up the FLEX boxes. And like many others who probably use the FLEX box from JB Hyperspectral, you end up standing in the field and you keep calling Tommaso and Andreas. And I'm surprised how patient they are. So you keep calling and you still get an answer, and maybe that would make their life a lot easier if the manual was a lot better. That's just a little bit of criticism. In any case, so this is what the setup looks like. The boxes overlook the forest. This is then in between the time when the boxes were installed, the weather got really, really bad. Several days of rain and the plane couldn't fly. And then eventually on June 13, I tried to magnify that quite a bit. They got their imagery. This illustrates the 2 FLEXes that we installed, one in an irrigated site that overlooks this irrigated spot and another one in this non-irrigated spot. So we have 2 FLEXes altogether. So this is an overview of some of the environmental data. Photosynthetic active radiation over here for this 15-month period from June 2024 to December 2025, just a few weeks ago, more or less, in temperature. And then we have relative humidity and VPD in this figure over here. And when you look at the VPD values, you can see that it gets, in the temperature values, it gets quite hot and quite dry during the summer month. This is May, June-ish something. There's a lot of, it's not as extreme as it was in 2025 due to the fact that there was lots and lots of rainfall, unusual rainfall in that area. The highway was closed. We tried to get to Zermatt. Zermatt was not accessible because the roads were flooded. And you see that in the data as well later on. So this is the effect of the irrigation and the control treatments. In blue, you can actually see soil water content in the irrigated plots, and here soil water content in the non-irrigated plots. Precipitation is also shown over here, and you can see that there's always a little bit of a fluctuation in soil water content at 80 centimeters depth. And there's one particular spike over here. This is April, I think 16, 2025. This is where heavy snowfall occurred in the Fienwald that actually led to ground damage in the trees. And this has a huge effect that you can see later on also in the leaf spectral reflectance and the solar-induced fluorescence data. Yeah, so just to emphasize the hot and dry peaks during the summer. This is the NDVI that we derived from the FLEX boxes, and you can see that there's some really, really low values. This coincides with periods when there was lots of snowfall. This is the point that I mentioned earlier, where there was a spring snowfall that led to ground damage. And I'm not quite sure if the drift that you see in the signal over here, if this is actually a drift of the sensor, or if that's a signal that is caused by a decrease in the canopy, because there was lots of broken canopy, and I think we still need to quantify that, or try to quantify that to some extent. I also threw in some chlorophyll, total chlorophyll data for the 2 treatments, just to illustrate that a little bit further. Down here is PRI and NIRV, and I think it's, for me, it's exciting to see how nicely the different leaf spectral reflectance indices are able to distinguish between the 2 different treatments, and that they capture the seasonality and the dynamics of all these processes quite well. So again, that's the heavy snowfall over there. So this is CIF-A and CIF-B, and apparently both can distinguish the 2 treatments quite nicely as well. What I would like to emphasize that it seems that CIF-A is consistently increased in the irrigated plots here in the blue shown versus the non-irrigated control plots, and that, it's dry here as well. The air is very dry. And CIF-B is less sensitive to drought. And the differences between the two treatments that are actually indicated by CIF-B are largely restricted to the summer 2025, to the period where we had actually peaks in vapor pressure deficit and really high temperatures. Okay, I had that already. When we plot CIF-A and CIF-B against VPD, this becomes even more obvious, where I think it's to me at least quite exciting to see that CIF-A is able to really well distinguish the two treatments, and that the effect is not as apparent when we look into the CIF-B signal. Okay, as I said, all this is very preliminary stuff. The pigment data, I have lots of carotene and xantho full cycle pigment data. All this came out of the HPLC last Friday. So this is really stuff in progress. But I think what you can see already is that there's an increase in CIF-A in the irrigated plots versus the non-irrigated plots. That the drought signal in CIF-A is pronounced compared to the CIF-B signal. And the next steps are to integrate all the leaf level physiological data that we have to validate and mechanistically explain drought responses in the canopy CIF in those drought stressed cotspine trees. Thanks, Todd.
Thank you, Ingo, for new data from a really well-maintained forest site in Switzerland. Other questions? Yeah.
Thanks a lot, Ingo. Thanks. You emphasized the after storm effect, or after snowfall effect, where there was a canopy damage, and you showed that in the NDVI. But I was wondering, in the other signals, in the near V or in the CIF, there was no, I didn't see any signal happening there, where I would have expected also the canopy source would change. Or the quick recovery that you saw also in your NDVI in less than a month, the NDVI was back to the original level. So what kind of damage are we talking about after the snowstorm?
Well, there are entire canopies of some of the trees that actually broke down. And like I said, we haven't yet quantified to what extent that had an effect on the signal. We have also an RGB camera that points in the exact same direction like the FLOX sensors or the CIF sensors. And the next step to do is just to figure out how much damage there was, and if we can quantify that and link to any of the CIF or leaf spectral reflectance data.
I didn't see it in the CIF. I didn't see in the CIF, or you didn't point out in the CIF, these peaks that you saw in the NDVI were not visible, or in an.
Yeah, I don't know. This is the first time that I deal with CIF data myself. From my point of view, there's a difference between the 2024 and the 2024 and the 2025 year. And before I say, oh, this is all the same, and everyone else tells me, oh no, this is not the same, I would be rather on the safe side. On the other hand, we have, like I said, we can actually quantify the damage, and I think that would make a lot of sense to figure that out later on.
Hi, thank you for this nice presentation. I was wondering if you have any LiDAR data at hand to characterize the different plots in respect to structure. Well, even though not in comparison over the running period, but maybe it would be also interesting to see if the different treatments already had an impact on the structure on the trees there.
So, as I said, the Fienwald is like an emergency room. There's a group who does terrestrial LiDAR, and there's also the person who flies the multispectral drone that has also a LiDAR system. So they map out the entire site, and there are multiple ways to quantify first the damage. And with regards to any canopy level structural changes, they have done a lot of. I think there are a couple of publications where they actually demonstrate and quantify how the irrigation and the irrigation stop had actually an effect on the canopy development. And in the current treatment, it's the trees from the irrigation stop experiments that developed a large canopy during the irrigation phase that suffer now the most from the water shortage in the site, and this is nicely quantified. I don't know the exact publication, but there's some work out about that.
Okay, good. Then, thanks again, Ingo. Yeah. And we come to the last presentation of this session, given by Nicolas Parazzo from JPL in the US. And if I understood it correctly, it's about a reanalysis of a large data set, which you will present. But the floor is yours, Nicolas.
Yes, thank you. And I don't want to be the person standing between a very long morning and lunch and pictures, but I promise I'll do my best to power through these slides. There's a few big points that I would like to make. I want to describe this land biogeochemical reanalysis that we've been developing to close the carbon, water, and energy budget, and the application of this to reconcile Earth observations, to benchmark Earth system models in advanced Earth science prediction and understanding. And then I have a bonus topic at the end if I have time. This is, I think, why a lot of us are here today. Earth's carbon budget uncertainty. We want to understand where the carbon is going and how that's going to change in the future. For most of the historical period, we've seen a linear increase in temperature with cumulative carbon emissions. And whether that linearity increases into the future or continues into the future or not depends on carbon climate feedbacks. And if we have kind of the dominance of CO2 fertilization, we might see a flattening of the curve. If we see something like more climate extremes and more perturbations to the carbon cycle, like we have been seeing in the recent past, we're going to see steepening of the curve. We don't really know, and we want to develop Earth system models to help us predict the future with more certainty. And so the question is, how can we use Earth system models to capture carbon climate feedbacks under changing forcing? The challenge is that ESM projections are uncertain due to state flux and process uncertainties. There's an opportunity to take advantage of Earth system model dynamics and benchmark these with the contemporary satellite program of record, which is quite extensive. We have NASA, we have ESA, and the other space agencies, so we have a wealth of data to work with here. The missing piece, though, is to be able to integrate all of this information together, the observations and the models to help kind of close the budget. And so we have attempts to do this, to assess our current understanding of the land, carbon, and water budget. Currently, these are insufficient. The carbon models, they do a good job. They're constrained by a small set of observations, but mostly they're still quite variable. They disagree in terms of the magnitude and the variability over time. And the same with water. It's even more variable. A big problem is that these two assessments are done independently of each other, where in reality, we know carbon and water are coupled, and so we should be developing these carbon water budgets together. So I mentioned we have just a massive set of Earth observables from all the different space agencies, including SIF, but also woody biomass, woody debris, snow cover, leaf surface area, canopy properties, carbon exchange for CO2 and carbon monoxide. We have water storage, runoff. We have all these observations. We also, there's a lot of uncertainties in these observations. There's a lot of inconsistencies in these observations. They're taken from different sensors, and that alone is hard to reconcile. There's big gaps in the observations. There are big periods that are just not covered. And there's also processes that are just unobserved, and we have to rely on models to capture those. One nice thing that FLEX brings to the party is that you have kind of a consistent set of measurements from a common platform. So you have SIF, you have leaf area, and you have land surface temperature. if we think about Sentinel and Flex being kind of a package deal. And so this is going to be an important, consistent constraint to have. We also have chlorophyll content, which we're trying to find ways to take advantage of in the model. And so just to kind of summarize, we have observations, a source of knowledge. It's a record of the past, present, and future. And we have a rich observation record, and we want to find integrated value. However, there's significant uncertainty and gaps, and we want to reconcile these incomplete observations. And I mentioned that some things are never directly observable, and so we need to provide that information using a model data fusion framework. And so now we have the models. This is an aggregation of knowledge over time, a diagnostic of the past and present. It gives us the ability to look into the future. And so we want to be able to combine the strengths across the different models. However, obviously, they're full of uncertainty, and we want to be able to gauge the individual performance of models. And some processes are way too complex to simulate, and so we want to be able to constrain a range of complex processes. And so observations inform models, models inform observations. And so the solution here is, as we're proposing, is a land biogeochemical reanalysis to bridge the gap between the two worlds. And so this is our model data fusion system called Cardamom. The ingredients to get this model going are kind of summarized here. We have the physical model called DALEC. We have parameters that we solve for using the observations, and those are given prior values, prior ranges, and so we're able to account for uncertainty based on those prior parameter values. We have the observation operators to link the model state to the observed state, cost functions to compare the two, and then Bayesian optimization to look for the optimal set of parameters that best kind of solves for what we observe. And so the reanalysis is going to be the way to integrate carbon, water, and energy, observations and physics. And so the goal here is that we are able to observe the state, we're able to observe the forcing, we're able to observe the change in the state, and we want to infer the function, the things that drives the interactions between all these different pools of carbon and water. And so this is kind of a summary of everything that goes into the model on the left. On the right is what comes out of the model. The things that are not in bold are what are observable. The things that are bold are the unobservables. And so we have states, fluxes, diagnostics, sensitivities. That last two columns are mostly things that are unobserved and very important for understanding how things are changing. And then we have carbon, water, and energy. So we have a kind of a complete closure of the budget and full accounting of uncertainty. And so this is probably the most interesting result that has come out of this reanalysis. We kind of a diagnosis of carbon climate sensitivities. And so what this is saying is that the carbon cycle is accelerating over time, over the past 25 years. And so the planet is absorbing more carbon, it's storing more carbon in both live and dead pools, but it's also releasing the carbon quicker. And so if we were to summarize this and think of Earth as a sponge, the Earth is becoming more spongy, which means it has more capacity to absorb carbon, but it also dries out faster, and so releases carbon faster. And so this is an important concept to think about when we're considering the effects of CO2 fertilization over time. And so the next couple of slides are less interesting, but still important. If we're going to have a benchmark for models, we have to have a full gap-filled model, a full gap-filled estimate. We need to be able to have long period of record to look at change over time, and we need to be able to provide variables that can be provided to modelers to diagnose their models. And so the idea here is that we're able to provide a gridded product. We're able to provide budget closure, so we include terms for everything kind of below ground and above ground and its change over time. And then we have kind of monthly resolution, so we can look at sub-annual types of variability, which gives us more insight into how things are changing at seasonal timescales. And more importantly, we also provide a reconstruction of the historical carbon and water observational records with full uncertainty. And so in this example here, we have flux observations for a decade, but we need to be able to extend that to the previous decade, and so the model data fusion allows us to do that. This example here is just showing that we provide uncertainty based on the ability to reconcile the model and observational state. This on the bottom right, this is monthly terrestrial water storage. You see a lot of gaps in here, and so this reanalysis allows us to fill gaps and then provide new data. This is ET. We don't have a lot of information about ET, but we are able to kind of, with a wide range of uncertainty, provide a time-resolved estimate of that as well. And this is just briefly an attempt to provide, to use cardamom as a benchmark for Earth system models. You see all the variables on the rows are all the benchmarks that we provide for carbon, water, and energy, and then diagnostics on the bottom, and then different models on the top going from left to right. The different colors are the scores, and you can use those scores to rank models and then project out into the future based on that weighting. And so this is a summary of the first 4 big points. I want to end with a kind of a reminder that coming from JPL, it would be remiss of me to not talk about the Orbiting Carbon Observatory. So we have this mission. There's OCO2 and OCO3. They were both designed to measure carbon with very high precision. And by having this very high-performing instrument, it's also able to measure solar-induced fluorescence. And it does this using the Fraunhofer line retrieval along the edge of the O2A band. And it does it with very high precision and stability. So we have this OCO2 mission, which has now been in space for 11 plus years. This is it's a free flyer. It's sun synchronous. It provides global coverage every 2 weeks. And it's the pixels are 1 kilometer squared. And so you get these really, really nice long-term repeated measurements over the entire planet. Then we also have OCO3. This is on the space station. It's the same instrument, and so the same type of precision. But it is, because it's on the space station, you get this precessing orbit. And so this means that sporadic, you get coverage from about 52 south to 52 north, but you also get measurements at different times of the day. And so you're never just measuring in the afternoon or just the morning. And so this gives you extra information about the diurnal cycle of SIF. And these are slightly coarser footprints, but still pretty decent. And so I just want to kind of provide an overview of what these things are observing and how they could be useful to FLEX. And so for OCO2, you see this kind of landscape scale measurement of SIF. You can see the gradients across land cover. You can reconstruct maps of SIF using machine learning, combining SIF with other measurements of vegetation to get these nice gap-filled maps. You can compare these maps to tower observations. We're using photospec, this proximal remote sensing, and you get nice linearity when you look at kind of things at monthly timescales. From the OCO3 end, this is an animation of coverage from OCO3 over Europe. Each day is a kind of a map of coverage. You see those lines are the nadir tracks, and those boxes, the boxy looking things, those are called snapshot area mode data. These are 80 kilometer by 80 kilometer boxes. They give you these really high density maps, and it's able to kind of point at any location that we give it. This is an example of a SAM over Cologne and Bonn, and so right outside the door. And so we have many maps that look just like that of SIF. And this is a map showing the locations that we currently measure in Europe. And so I just want to kind of bring this to the table as something that could be helpful for doing CalVal for FLEX, but also something that OCO2 can learn from what FLEX is doing. And I'm sorry I went over time.
Thank you, Nick. Any question for Nick? There's one there.
Hi, just a quick question about the future of the OCO2 and CO3 missions. Are they staying? Because I heard that they might be taken down.
So great question. You kind of teed me up for that. Yeah, so we're actually trying to extend both missions 3 years. We're in something that we call the senior review. I am a part of the senior review. We are really trying to push the SIF side of the missions and de-weight the carbon side of the missions because of our new reality back home. And so we're working on selling the missions and making our case. We're gonna go to NASA in April and present our case. At the very least, we're gonna be able to continue this through September. After that, we won't know. So it's a waiting game, but we really think that we can make a good case with SIF because we're all about trying to do applications in agriculture and forestry, and I think it's really valuable for that.
Question from Christian.
Hi, thank you for your great talk. Oh, it's okay?
Yeah, go on.
Okay, thank you.
Oh, there he is.
Yeah, this question is not about the scientific things. I'm just curious about your opinion. Then, if the cardamom, the accuracy over the 99% or 100%, then what we can do by that Earth system model?
You're asking if we get to a point where cardamom is 100% accurate?
Yeah.
Then we don't need Earth system models. No, I don't know. That sounds like a fantasy, but if we have a world where we have complete truth and no uncertainty, then it would be very easy to figure out which model is right, or it would be very easy to probe the model and understand what's going wrong and make the model better. So that I don't know, that's a nice hypothetical situation, but...
Okay, I see. I'm just curious about your opinion.
Yeah.
Thank you.
I have a question, actually, on your residence time in... how are you sure that it's diminishing, and how are you not sure that your respiration is due to more carbon in the soil, and how are you validating that?
Well, so we have the above ground and below ground biomass, and it appears that there's less carbon staying in dead biomass and more in the live biomass. And so it appears...
This is modeled. This is not observation.
Well, it's constrained by observations. And so we have the Harmonized World Soil Database providing one constraint of soil carbon, and then we have 20 years of biomass data from ISAT and JEDI, ISAT and JEDI. And so it may not be the case, but this is what it's suggesting, that this is the reason why we get increased turnover.
You had a question as well.
Yes. I was wondering if JPL already has been communicating with the FLEX community for maybe some, during commissioning phase of FLEX, some pointing of Sentinel-3, pointing of OCO3 to some interesting areas.
Yeah, that's why I'm here.
Oh,
yeah,
we should
definitely talk about that. Yeah, I mean, that would be amazing. I'm surprised that we haven't done that yet, but I think there's more interest right now to definitely do that.
And it's still in time for the...
There's still time. Well, yeah, so there's plenty of time. We can, add a new SAM now. We can kind of start planning now and hope for the best. The space station, it's already there. It would cost them more money to decommission it. So we think it's going to be there for another 3 years. Nothing's guaranteed, but it would be weird if it didn't happen.
Europe is going to pay big.
Okay, thank you very much. Let's have a round of applause for all the speakers. And I will invite everyone to come forward towards the screen, which will probably change to a flex. No, sorry, Nick. We're ending here. And yeah, can you imagine?
Can we please have the background, the graphics? Yes, exactly. Thank you. And we will restart at 2:30 sharp, so please try to be in time for the next session. Thank you and enjoy your lunch. Good afternoon, everyone. We are about to start, so please take a seat. Okay, good afternoon again, and thank you for being back after lunch. This session that we have is a sort of joint venture between two short sessions, or with two subtopics. It's about novel products, or products which are in some case slightly outside the primary objective of the mission, but certainly extremely valuable in perspective, and which have been explored not just in the last couple of months, but have a long history also in the mission and in the history of these workshops. So it will be in total 5 presentations, basically 3 focused on water and 2 on novel products. And then we have 15 minutes at the end. So if the speakers stay within the about 12 minutes, then we have still time for some direct questions, and then we have we want to hear from you in terms particularly of novel applications and scientific products that might be of interest for FLEX and could be explored in the coming years. So with this, we will start with Alex Dam, now familiar face also during the last days to all of you, but on a different topic, specifically on inland water research related to FLEX data. Please, Alex, floor is yours.
Okay, thank you, Marco, for this nice introduction, and yeah, welcome to after the lunch break to this presentation. There are a few things different compared to the sessions we had before. First of all, there's less audience. I guess those who are not here, they will for sure miss all the nice beaches, beach photos, and so on. So second, it's about water, but aquatic systems, and also a big difference compared to my presentation from last time. We see much more blue pictures instead of green pictures. So I'm talking about some research we did, or results we did, in relation to the question how FLEX vision data can maybe help to advance aquatic research. And I would like to start with a few words about aquatic systems. So they are quite important, and I guess you're aware of many of the services they provide. They are responsible for climate, controlling the climate. They are involved in the cycling of carbon and water, for example. They provide fish, very important food source. They also, their paths or transport paths, basically. And many of my colleagues enjoy sailing, so they're also good spots for recreation and obviously also biodiversity hotspots. Despite their importance, they are largely unknown. And one of the big problems we have here is that there is water. So we cannot live or stay longer times underwater. We even have challenges to stay on the water in combination, if there's water in combination with wind. So the information we have so far are basically coming from point measurements. We have a few lakes and waters, open waters, oceans, coastal areas equipped with intense equipment, but these are point measurements. There's a high spatial temporal resolution at times, but many things are completely unknown. And here, in order to provide information, remote sensing plays a very important role because we can do the spatial temporal mapping. If you talk about remote sensing of aquatic environments, there are a few things to consider. And for colleagues familiar with this topic, this might be a bit too simple what I'm telling now, but I think it's very important because the radiated transfer we are dealing with here is a bit different to what we typically used in when we work with vegetation. So I would like to go a bit into the details, scattering, absorption, and so on. If you talk about scattering, we must say it's quite complicated. First of all, we have the specular reflectance or sun glint can come from the sky or sun. And the problem with the specular reflectance, these are these white spots you can see in this area here. This is light reflected at the surface and is not entering the surface and does not contain any absorption information. This is a problem. It's kind of a disturbing signal we have to get rid of. And then the transparency, so how large we basically, how large deep we can look into the water depends on the transparency. Talking about absorption, water absorbs a lot of light, and with the result that we have a very low signal to measure. So the water leaving radiance is typically really small. And then, because water absorbs a lot of light and almost all light beyond 700 nanometer, we have a very small or narrow spectral window we can work with. If you think about multispectral data, Landsat, Sentinel-2, there are only a few spectral bands we can really use, so spectral independent measurements we can use, and this also limits the number of independent observations we can get from water. So and then in terms of visible things or invisible things, so there's a lot of things going on in the water, a lot of constituents and contaminants. Some of them are partially visible, for example, dissolved, suspended, organic or inorganic material. But other things we are interested in, for example, water quality parameters like pH value or so, or some certain pollutants, are sometimes or quite often invisible because they do not interact with light in terms of scattering and absorption. So this makes it quite challenging and also narrows down the number of information we can infer from remote sensing of aquatic systems. Another issue is related to the variation. Water is really variable, in particular aquatic systems. We have very large fragmentation and also in very large spatial diversity in terms of the size of the water bodies. Then next, we have very pronounced vertical variability. And depending on the turbidity of the water, we can assess certain parts of it, sometimes not. And water, or the things in the water processes, in the water constituents, or the presence of constituents can be highly dynamic. So they can change quickly in minutes to days. And in case we are talking about extreme events, for example, flooding events, or so, we also have to deal with high spatial temporal variation. These two figures here I would like to highlight. I was not involved, but this is from colleagues at EARWAG, data lakes and ALP lakes. These are very interesting tools, platforms, web-based platforms to get access to data. EARWAG and other colleagues are measuring in Switzerland, ranging from in situ observation satellite data up to A1 and 3D modeling results. So very interesting if you're interested in this. So there are a lot of challenges related to the question of how can we measure how can we use remote sensing to assess aquatic systems. Now let's lose a few words about the observation capability we have so far. And I'm only addressing optical missions here, and this is a kind of personal judgment in relation to the spectral, spatial, and temporal capability we have in space to address or observe these waters. And this obviously is a small selection of available satellites, but they are kind of role models for other similar missions. So if you, for example, look into the spectral capacity, we can say Landsat Sentinel-2 have only a few spectral bands, so there's some limitations. And for Sentinel-3, there are more spectral bands, they're optimized, but still it's not as good as, for example, hyperspectral missions like CHIME, PRISMA, NMAP, or dedicated water missions like Glimmer or PACE. So then if you have a closer look at the spatial resolution, we can say, okay, Landsat Sentinel-2 offers 10, 20, 30 meter resolution, it's quite good. But then the other missions which offer high spectral resolution, they suffer from a low spatial resolution. And then last but not least, temporal resolution. I told you already that water is really dynamic. Here, PACE, Glimmer are quite good, offer good capacity, but others, for example, the hyperspectral missions we will have soon or already have, they are limited. And you have to keep in mind this is only for inland waters. I'm not talking about oceans here. So this brings me to my point. There is large observational gaps. In order to better address aquatic systems, we need dedicated water missions. And FLEX is obviously not a dedicated water mission, but provides opportunities. First of all, it's complementary in terms of spectral resolution, offers very high spectral resolution. This can be, for example, used to advance product development scheme, the development of product retrieval schemes. And it can be also used if you have an observation to, for example, demonstrate what can be done with dedicated water missions because of the high spectral resolution from FLEX. So this was an introduction motivation, and now I would like to give you a quick idea or two examples on things we did where we think this could be something where FLEX data could be also used in order to bring these studies forward and move from the in situ studies basically to maybe larger scales. So example number one is related to fluorescence. In this particular case, we aim to derive fluorescence from hyperspectral data and use this as a proxy for biomass. SIF per se, if we relate it to phytoplankton, can be used for biomass estimates, so how much phytoplankton is there, but also health status and photosynthetic activity. And in our case, we wanted to see how SIF can be related to biomass or chlorophyll A estimates eventually. And therefore we use data from a profiling instrument, the so-called TETIS profiler at the FLEX-Fluor platform in Lake Geneva. And this instrument goes up and down, it takes roughly 2 hours for one profile, and I think up to a depth of 50 meters, and then is measuring a lot of different things. Optical measurements, inherent optical properties, apparent optical properties, and so on. And from these observations, we then, using a model inversion scheme, so we basically parameterize with the in situ observations, hydrolite, and then compare this with our optical measurements, we could infer the diurnal variability and depth variability of fluorescence. The signal looks a bit different to what we saw in the last presentations. It's just the first peak, and it's also related to the fact that water absorbs above 700 nanometer. But we can see a dependency of the SIF signal in relation to depth, because if you go deeper into the water, there is less light available, and also has a diurnal variability. Okay, so far so good. What we then typically do is we take the SIF measurements as a proxy. So this is one of many approaches for chlorophyll A. So the interesting thing of hyperspectral data is that we can use many ways to assess chlorophyll A. We can, for example, make an empirical link between fluorescence and chlorophyll A, or we can also use the absorption properties of chlorophyll A. So 2 different strategies at least. And in this particular case, we compared chlorophyll A estimates based on SIF and absorption spectroscopy. And we could find some quite unexpected behavior. What we found is that with depth, there was lower depth, there was an increasing divergence between both chlorophyll A estimates. And this was the case in the morning, midday, and in the afternoon there was no problem. And then we thought about this and investigated this further, calculated some correction factors in relation to light availability, and so there seems to be a dependence on light. And by further investigating, we figured out it was basically NPQ, non-photochemical quenching. So this means eventually, if you want to use a chlorophyll SIF estimate, in order to predict chlorophyll A, we need to know if the photochemical or non-photochemical pathway dominates or not. Otherwise, you're getting wrong results. The second example was about the use of hyperspectral data to assess phytoplankton diversity. In this particular case, we have another lake, Lake Greifensee. There's a floating platform with a spectrometer on top. And then colleagues from ERWAG developed an underwater microscope. And this microscope is taking pictures at a depth of 2 meters, and then with artificial intelligence can basically classify the images and derive the abundance of certain species. We use this as a reference. And then we took our spectrometer measurements from a WISP station, similar to a FLOX, but more optimized for water measurements, and parameterized VASI, a radiated transfer model, inverted the model, and came up with time series of species abundance. And this was then compared. And here are the results. What we can say is that for 3 different phytoplankton species, cyanobacteria, green algae, and diatoms, you could achieve quite good results. We could nicely track with our optical data the blooms. For diatoms, it was a bit more, for cryptophyta, sorry, it was a bit more difficult. We also did an analysis to figure out the capacity of FLEX. This recent work from Loe. Just a quick explanation what we see here. This is basically the capacity or the accuracy with which we can retrieve a certain concentration of a certain phytoplankton species. So here you have the phytoplankton species, or let's say here, and then different concentration levels. Yellow means perfect, violet means it doesn't work. So and in the first column we can see the reference case, and everything for all sensors should look like this. And for PACE, a dedicated water mission looks quite good. For FLEX as well, although it's not optimized for aquatic remote sensing. And even for certain species outperforms, for example, Prisma and Nmap data. So these are good news if you, for example, want to use FLEX data for phytoplankton species assessment. A few concluding remarks. FLEX will support the aquatic community, and we are really thankful for this, because there are dedicated observations of inland waters and coastal regions. There's a dedicated mask, and the data will be acquired. And we think that the high spectral resolution of FLEX offers new capability, for example, to retrieve new products or refine the retrieval of certain products, full fluorescence emission spectrum together with MPQ, hopefully. With this, we could then maybe constrain chlorophyll A estimates via SIF. And we can also use this data for phytoplankton diversity assessments. There are just a few papers which give you more insights into these conclusions here. And last but not least, FLEX can also offer interesting observations and insights if we are aiming to develop dedicated water missions. In certain regions in Switzerland, we might get maybe 1 picture or maybe three for observations a year because of cloud coverage and the repetition time of 27 days. But still, these observations might be really valuable in order to show what we can do with this data, and then maybe provide this information or demonstrate what can be done for new missions like Galini or something like this. So the pro of FLEX is the high spectral resolution. The downside is a bit the low spatial and temporal resolution. Okay, that's it from my side, and thanks for your attention.
Any questions from the audience?
I've got one related to the last figure you've shown with the potential of the different missions. This is only taking into account the spectral, the availability of spectral information, right? It's not taking into account the spatial resolution. So you are assuming that the different species are uniformly distributed over a 300 meter pixel and over a 20 meter pixel, and okay, thanks.
I also have a question on the spatial resolution. Is there a difference if we talk about coastal waters or inland waters, where I can understand that the spatial resolution is needed, if it also accounts for, if I think about open waters, so open oceans, where I would assume that they are better mixed?
Yeah, I think it's important to differentiate a bit better. Unfortunately, time was not sufficient. So my focus was more on inland waters. Obviously, for open ocean, the distribution of the constituents and algae, and so I think the 300-meter pixel size should be sufficient, because Sentinel-3 was also developed and optimized for these kind of ecosystems. But inland waters means we also have small lakes and ponds. We have rivers. Sometimes there are some trees above the river. So, and there can be also, if you have an inflow in the delta region, some inflow of sediments from rivers or so, in order to track this variability, then this 300-meter resolution can become a bit, it's not sufficient. It really depends on the application and the inland water type you would like to, or you would like to investigate, yeah. But we cannot say FLEX is the mission offering the perfect spatial resolution. Spectral resolution, yes, but spatial depends on the investigation you have in mind.
Thank you very much, Alex.
Thank you, Alex. Our next speaker is Tristan Amel from and is going to speak about 5FLEX.
Hello, everyone. So thank you for having me here. I will present the 5FLEX project, which is dedicated to any kind of water, actually from oceans to coastal waters and a big of the lakes. And here it's investigation of physiology and diversity of different species of phytoplankton through the synergy of FLEX and Sentinel-3, as I will try to demonstrate. And here we will have a focus, on a very maybe tiny focus of the project, on the top of the atmosphere simulation, including the sun-induced fluorescence of the phytoplankton. Okay, so I would like to acknowledge all my co-authors listed here. So it's a very nice consortium with different institutions from France, Italy, Germany, Norway, and with ESA. Okay, so starting with phytoplankton. So here on the right side, you can see the minute things which are the phytoplankton. So it's very diverse, very different optical properties, different functioning in the environment, different marker of the biodiversity. And one of the achievements of optical remote sensing was to get insight of the concentration of those phytoplankton within the global ocean, and also to compute the primary production. And for the first time, thanks to satellite, we had an estimate of the primary production of water, which is kind of equivalent of the primary production over length. Okay, so from passive optical remote sensing, what we can have. So first, as you can see here, we need to sense aerosols, because aerosols are in between the satellite and the water, so of course we need to consider them. Sometimes you can have coupling of the aerosols that can feed a little bit the phytoplankton as well, so it's something important to have in mind, that we need also the aerosols information. So concentration, primary production globally, as I just said. And if you zoom in a little bit, you can also have very nice views for water quality monitoring for inland waters, coastal waters, and also open ocean. But here you can see in an estuary, if you get the image and you process with all the algorithms, you will get chlorophyll A, which is a proxy of the biomass. Suspended sediments, you can have also dissolved organic matter, transparency, different kind of parameters like this. And another very useful tool from the remote sensing optical system is to sense the different species of phytoplankton, because sometimes you get harmful species which is a threat for society or even for fisheries, etc. Okay. So that was a picture of what we can achieve from actual regular optical satellite for open ocean and water. And here in this project, we want just to know what we can achieve from this kind of synergistic approach by coupling FLEX, which has a very high spectral resolution, but we will miss many wavelengths actually, that is useful for water quality monitoring. And we will link to a wider spectrum, but very not resolved as FLEX, to get insight of the blue bands and also to the SWIR bands to make atmospheric correction, aerosols monitoring and stuff. Okay, so coming back to the phytoplankton, so that's this tiny stuff here, but actually that's the main driver of everything, because that's the first element of the food web network. So it will get a bit of gases like CO2, a bit of nutrients, a bit of photons, and will build up new organic matter like long chain of carbons that will be recycled through the food web, and finally, eventually, part of this carbon will just sink into the depths of the ocean. It's what we call the biological carbon pump, okay? But this biological carbon pump is controlled by what we call the photosynthesis, which is very complex, and that will depend on the different species, but also on the photophysiology of the cells. For example, if you have a lack of iron, if you have a lack of nutrients or too many sun, everything will be changed. But this change, this photophysiology, could be observed through the fluorescence. So that will be the goal of the project, is really to get the fluorescent signal to inform about the state, physiological state of phytoplankton. Okay, just to get into a bit of details here. So we have the reflectance simulation, and we change for exactly the same water. So we have exactly the same water, same phytoplankton concentration. You will just change what we call the quantum yield of photosynthesis, and you will see that the reflectance will change. And this information is a complementary information that we miss a little bit for the moment, and that could be achieved through the FLEX mission to get this small bump to get the physiology parameter of the phytoplankton. And by the way, if you see this equation, so if you get the primary production, like very crude assumption, approximation, you have to get the quantum yield, you have to get the absorption of a phytoplankton, which is dependent on the species, the biomass, and the irradiance. Okay, so just basic summary of the project. With all the team, we have different work packages. One is dedicated to see what kind of region of the world we have to prioritize to make a new acquisition mass. So I have just one slide, I will go quickly on it. And afterward, it's all the chain to provide simulations at the top of the atmosphere to afterward proceed with algorithm development, validation, etc. And here, in parallel, we have also deployment of different instruments, including the FLOX, that will permit to validate a little bit all the theoretical approaches and also to validate the algorithm implementation. Okay, so just about the acquisition mass. So what we did is to have kind of information-driven acquisition mass. So we got a lot of different data sources to see where fluorescence can be very important to monitor. So we end up with seasonal mask, and here you have like the static masks that could be, that give you actually the big picture of what we want to have FLEX data. Okay, so coming back to our story of the phytoplankton, we have two different scenarios. One is open ocean, when you get oligotrophic to blooming conditions with this kind of spectra, actually. Here, that's our effort to simulate the fluorescence, but I will get into more detail later on. And the second scenario is more in coastal waters, or when you have big blooms with lots of algae in it. And here, the goal will be to identify what kind of species we have. Okay, so to do that, we need simulations at the top of the atmosphere. So we provided of another project we have with ESA called RadicalNet. And this project was updated with a component for ocean sites. So what we did is to get in situ monitoring, in situ measurements at sea, and propagate it at the top of the atmosphere to proceed with calibration validation exercises. So we use this code to do everything, but I will try to give some methodology before. So what we want to get or to measure from the sea or from the satellite is this one. So it's a tiny signal. That's the water leaving radiance, okay? But actually what we have is this one. We have, okay, the skylight, so in blue here. We have absorption due to the gaseous absorption in the atmosphere. We have scattering. We have reflection, transmission with Fresnel stuff. And everything will be convolved at the end, and we need to disentangle everything to get, okay, this one. But here, to proceed with the simulations at the top of the atmosphere, we start with this one, and we have to compute all the others. To do that, we need to have a good radiative transfer modeling, and particularly above water, you need to include the polarization nature of light. Actually, when you include this polarization, you need to go with vectors, matrices. But once you have that, you need to feed, the equation with actual parameters such as absorption, scattering, phase function, or phase matrix. You need to get for the atmosphere and for the water. The second thing is to have a very nice coupling between the atmosphere and the ocean, because here you have a lot of reflection, as it was said by Alexander before. So you need to take into account polarization. So here it's still a matrix, so it's a BRDF, but it's forming a matrix to get all the polarization terms. And the same if you want to deal with the transmission. To handle that, we use a code we developed several years ago called OZOAA that can do it, but actually it's not done for hyperspectral simulations. So we coupled these simulations with the Libratron package to get very high resolution outcomes of our model system. Okay, so first thing is to have a good representation of gaseous absorption, as I said before. So here you have the full range, because we want to deal with FLEX, but also Sentinel-3, as I said. And here you have a zoom in of visible to near infrared. And here you see your main guys in green, which are actually the oxygen bands of interest here. So once you have everything, you can put different kind of aerosols, you can put different kind of viewing geometries, etc., and you output the reflectance or the radiance in radiance unit. But here there is no phytoplankton, so we need another thing very useful to do is to compute the downwelling irradiance at the bottom of the atmosphere, which is needed to compute the fluorescence. Okay, I will try to speed up it a bit. And here we are very fortunate because we have a nice database we collected over the years for different kind of phytoplankton with my colleagues cited here to get actually the ID of the different, the optical ID of the different phytoplankton in terms of scattering, absorption, etc. And we have also very nice measurements of the fluorescence in vivo done by Rüdiger Rödgers. And here you can see that the fluorescence is very different than what you have in the vegetation part. Once you have everything, you can compute the reflectance for any kind of concentration, any kind of species. You can add the fluorescence or stay without any fluorescence. And you change. So here the color are different phytoplankton species, and here different concentration. Okay, but what we want to have is something at the top of the atmosphere in the configuration of FLEX. So the last step is to make the convolution with the spectral response of FLEX. So we got everything from ESA to do these simulations. So here just to see how the transmittance can change if you change the spectral response. And you ended up with this kind of graph. And for us, it was the very first time we saw this kind of spectra with a peak due to the fluorescence, because usually what you use as instruments in our community is this kind of spectral resolution. And with this kind of spectral resolution, you have no peaks whatsoever. So here it's very interesting to see, we have a new way to see the information about fluorescence and maybe to decouple fluorescence and elastic scattering. Okay, so you can do any kind of concentration, and at the end, you can propagate it at the top of the atmosphere. So here you have in colors different concentration in phytoplankton, in radiance and in reflectance unit. And just to close up the presentation, here you have different species simulated, and on top of this, on the FLEX simulations, you have the Sentinel-3. And here you can see as a dot, and here you can see how many more information you will have from FLEX than Sentinel-3. Okay. So just to end the presentation, we have everything, you know, stored in NetCDF format. You can play with Jupyter Notebook if you want to go a little bit into the analysis of those data, of the simulations. And here my conclusion, it's a very start for me of having this kind of new insight of the fluorescence through the passive optical radiometry. So now we have a very nice work for developing inversion algorithm to retrieve sun-induced fluorescence and different optical and water parameters. And here it's also the first deployment, or maybe not the first, but very large deployment of the flux sensor with many other instruments to characterize the performances of this instrument for water purposes. And I will have to thank you. I'm a bit late, and here just an image of the notebook, if it works.
Thank you. That was great. Are there questions? Short question? Yes. Yes.
Just on the acquisition mask, how big are the differences of what you would like to have and what the baseline currently with Flex is?
That was the baseline?
Yes.
But here sometimes we will shrink, sometimes we will enlarge. So yeah, the goal is to maximize, you know, everything, and also the cost of the acquisition. So that's why, if it's not interesting, we don't want to have images, or we can, you know, get more interest to the other one. But just to let you know, to have the picture in mind, it will be like this. Okay.
Any comment? Otherwise, thank you very much again, Tristan, and we'll move to the next speaker, Anna Göritz from University of Freiburg in Germany, and we stay on the topic related to the validation of aquatic products for FLEX.
Yeah, thank you. Hello, everyone. My name is Anna Göritz. I'm a postdoc at Freiburg University, and I'm working there as a PI in the EcoSense series on forest remote sensing. So in case you have any questions about our fluorescence measurements or validation plans there, also feel free to approach me. But I'm also having an interest in aquatic remote sensing and a bit of background on that as well. So this is what brings me to this topic here today, in which I would like to introduce to you the AquaValex project. So, can I? This project was submitted and endorsed in the framework of the announcement of opportunity from ESA. And the aim is to validate and extend products from the FLEX mission also for inland and coastal waters. And this includes basically the ABC category, so the direct comparison of satellite-derived reflectance with simultaneous in situ measurements, and indirect validation through inversion of radiative transfer models, and also an intercomparison of FLEX products with products from other sensors. So this, in this project, we're a group of currently 4 German research institutions, being the Helmholtz-Zentrum Herian here in Germany, Alfred Wegener Institute, the German Aerospace Center in Oberpfaffenhofen, and Freiburg University. And the project is led by Martin Hieronymi, and unfortunately he can't be here today, so this is why I would like to give you a bit of an overview what we're planning here in that project. So here on the right-hand side, you can see a bit the work packages that we would like to cover in the framework of 3 to 3.5 years. So the idea is to, well, bring together our expertise and techniques to set up or establish a comprehensive in situ reference data set. And of course, also look into then the data analysis and the retrieval of actual level 2B products. And finally, also look into opportunities and capabilities to establish a permanent ground truthing station in an inland water here in Germany as well. So let me start a bit with the planned activities that we have outlined. So basically, they can be divided in 3 categories. So we have the short-term campaigns that are mostly to enhance probability of matchups. So that will be rather individual activities where we just go out forecast-driven by taking chance of flexible small boats, UVs, and so on. So this is rather rapid, spontaneous campaign, so to say. But then, of course, we're looking into midterm planned campaigns where we then come together, coordinated with FLEX overpasses to hopefully have some in situ validation in larger lakes, inland or coastal waters. So this could be, for example, at the Lake Constance or Lake Banta or Bad Zwischenahner. This is all places where we already have gathered some in situ measurements and then can build up on that as well. And then the third category would be like long-term planned missions, so to say. This is largely organized by Alfred Wegener Institute and Heimatzentrum Herion, where there are several expeditions laid out for the next years. These cover different areas and regions, and of course here's also the opportunity to look into additionally the fluorescence products and extend a bit, or this can then be combined with an extended parameter set and also set into a broader scientific context here. So this is about, more or less the types of campaigns we're planning. For all this, we can draw on a lot of experience, but also equipment that we can make use of. So we also use some custom-built, specific tailored instrumentation for aquatic remote sensing purposes. So as it was nicely introduced in the previous talks, there are some specific requirements when it comes to aquatic remote sensing in terms of low signal analysis, also making use of smaller wavelengths, not having that information in longer bands and so on. So for example, one example would be optical spectrometric system developed at DLR for handheld reflectance measurements, or the Limnovis RV vehicle that measures simultaneously above and below water and also retrieves a bit of, for example, benthic information and bathymetric parameters and so on. So from Freiburg University, we will contribute with drone observations, and all these measurements take place in situ. But of course, you have to imagine that this also comes with a lot of laboratory analysis, for example, pigment analysis, water constituent characterization, absorption, scattering properties, and so on. So this will be carried out in the laboratory, of course. Here you can see the simulations for different concentration ranges of chlorophyll with a combination of several phytoplankton groups that can be, even though when having a small single, you can still differentiate a bit what is actually going on inside the water. And this is a simulation where you can see here at low concentrations, of course, there's no signal, and only when it comes to really high concentrations, you have here in the red and red also signal contributions, also with scattering particles then enhancing that signals there. So typically when making use of this remote sensing reflectance above water, when it is corrected for atmospheric effects and so on, we make use of the pigmentation shape, also the well, basically, usually the absorption and scattering properties of everything this is in there, but in the framework of the Aqua-Valex project, we would like to place a particular focus on fluorescence optical properties, of course, as well. So in order to include that into the retrieval algorithms, as shown in this example here for cyanobacteria, where you have the fluorescence mission, not only the chlorophyll, but also with accessory pigments on top. So this is a slide from Astrid Bacher's team. When it comes to actual level 2 product retrieval, we will make use of different radiative transfer tools, and this will be Hydrolite, the watercolor simulator Lavazi, but also skiatron modeling. And this is an example from Astrid's group where she already some time ago demonstrated the retrieval of fluorescence from skiamaki data, as shown here on the left-hand side. So of course, this can also be built up for FLEX, and some groundwork already has been laid out with respect to creating lookup tables based on skiatron, and also phycobilin retrieval, which falls into the above 500 nanometer range, which is of course relevant for the FLEX mission. But this is already like promising groundwork, and here is the retrieval also demonstrated with another tool, which is the VASI watercolor simulator by Peter Geger. And you see here on the left-hand side a scene from the ANMAP mission. Here you can see a coastal estuarine area where you have an Aranet OC station in here. And now you can see on the right-hand side the pixels, the reflectance spectra around this station. And now you see what we have to deal with in an aquatic environment. First of all, the reflectance values are really, really low. Then we have, as also Alex introduced, the strong glint contribution here, which is depicted here as a bright curve. And once you correct for that, then your actual reflectance is here in the realistic range. And here you can see a nice matchup also with the data coming from the Aranet OC station. So once you have these nice curves here, you can actually retrieve the water constituents concentration. In that case, it was chosen to be done of flagellates. You can take this as chlorophyll concentration for that example. And also you see total suspended matter here on the right-hand side. And then in the next step, this is something really preliminary. We now look into fluorescence retrieval. And this is something Peter Geger just demonstrated, that he actually calculated the fluorescence quantum yield out of this retrieval by using a Gaussian approximation for the fluorescence. And then you come to a representation like this, which is already really promising in that way. So of course, we would like to build up on that, and this in principle can be also extended for additional pigmentation. So nice thing about the FLEX mission is that it will fly in tandem with the Sentinel-3 mission, and this will allow for a broader view, but also for like a benefit for atmospheric correction, as shown here from Martin Hieronymi and his team. Here you see indicated the FLEX tracks, and you see the North and Baltic Sea here on the left and right hand side in the center. And you already by eye can maybe distinguish a bit differences in the coloring. If you have a closer look, and this is what the team there around Martin did, and Rudiger, he's also part of that. He is here today. Where is he? Hidden in the back. Okay. What they found is that you can actually really nicely, based on the Sentinel-3 data, classify the different optical water types here. So this also helps in terms of data interpretation and also enhancing the retrieval algorithms if you have a prior classification, for example, that can reduce the uncertainty, and they also help in interpreting future upcoming FLEX data maybe. So a final work package that I would like to focus on is what we would like to work on is the permanent calibration validation station in a lake here. I'm also really open to exchange on that. So the idea, as I said in the beginning, is to have some automatic continuous ground truthing. And of course, there will be a lot of exchange, but we also need to perform some measurements to really define the specifications and to decide on the combinations for additional measurements with respect to atmospheric parameters, in situ measurements that can be performed simultaneously, and with the idea to then finally have an implementation roadmap. So all of this, of course, needs to be realistic. You need to find places where this is applicable. You need to find also the maintenance operation capacities and so on. So this is part of the last work packages of the AquaValex project. So with this, I hope I have given you a bit of an overview of what we're planning to do here. So we would like to build up on all the experience on IOP characterization, hyperspectral sensing hardware, radiative transfer modeling, the experience with calibration, validation, data collection, with the idea to really set up a nice, comprehensive in situ reference data set across different water types, and then work and push forward more also the retrieval of level 2 products and extend what is currently there, and finally also look into automatic permanent calibration validation sensing in Germany here as well. So thank you for your attention.
Any questions for? I actually have. Yeah.
Which is the
microphone, please?
Yeah, just a curiosity. Which is the magnitude of the peak position that you show in the map?
So
Okay.
which?
Yeah, no, on the right side, yes.
So here you have
No,
like
the magnitude of the peak maximum.
So actually it's hidden here. It is also something maybe it's interesting to understand that in aquatic, you can see it a bit here. Well, even though if you have here, so this is not like chlorophyll fluorescence effects, for example, this peak here, but you have your spectra are largely shaped by the absorption of chlorophyll and the absorption of water that comes on top. And then from the remaining what is there, you still need to fit in this small peak, which then reflects the fluorescence emission in red. So, yeah.
I think there are quite some things still to bridge between the two communities. For example, also the how, in which units we express fluorescence, because I think, like I've seen it in reflectance unit most of the times, right? In your case, well, there's the terrestrial SIF community is used to fluorescence in radiant units. So I think we still have some harmonization to do in terms of understanding each other.
Well, of course, it is calculated in radians, but you need to convert it.
Yeah, clearly. And I have a curiosity on slide 10, if you... Yeah, because you say here are the FLEX tracks. What I mean, clearly over time, then FLEX covers all the... covers globally. So
well,
I just wanted to understand what you meant by that.
Actually, a good question. The idea was to indicate a bit that we have a broader coverage from Sentinel-3 that is measuring in parallel, and that from that we can have a combined optical water type classification and atmospheric correction, which then helps to have this simultaneously recorded FLEX data being interpreted.
Thank you. Any final questions? Well then, thank you very much.
So we will now move to next session. So thinking beyond the state-of-the-art novel applications for FLEX. And there we have two presentations. And the first presentation will be given by Jennifer Johnson from University of Kansas.
Thank you so much for the introduction and for the opportunity to speak today. Before I start, I want to acknowledge I'm a new faculty member at the University of Kansas, and I want to acknowledge my new group there, as well as a broader network of collaborators who've really helped to push this work forward. And most importantly, I want to acknowledge Joe Barry, who's been a partner in sort of the conception and execution of what I'm going to talk to you about today. The title of my talk is also the title of a contribution from my group to the CalVal AO. And in my talk, what I want to do mostly is to motivate what we're interested in doing and to tell you about some of the background and and preliminary work, and then I'll end by telling you about the project proposal. So here we go. The theme of my talk is going to be that the concept of the maximum photosynthetic capacity of terrestrial plants is something that could really be considered to be central to the FLEX mission. And there are two reasons for this. One of them is that photosynthetic capacity is a variable that mediates the relationship between the environmental drivers of photosynthesis, the amount of CO2 fixation that happens, and the chlorophyll fluorescence that comes out. So understanding photosynthetic capacity is important for understanding what chlorophyll fluorescence means in a quantitative sense. Photosynthetic capacity is also interesting because it's modulated on ecological and on evolutionary timescales. There's lots of variation between different plant functional types or different evolutionary lineages. And of course, it's modulated also by environmental resources and stressors. To date, sort of a short summary would be to say that the plant and earth sciences communities have really tended to understand photosynthetic capacity for terrestrial plants through the lens of the Farquhar, von Kammerer, and Berry model of photosynthesis. And when Christiane and colleagues introduced the representation of fluorescence into the Farquhar framework, the underlying concept of maximum photosynthetic capacity that had been present in the original Farquhar model was really preserved and imported into the new scheme. This is, of course, the idea that the enzyme Rubisco is the only thing you need to think about if you're trying to quantify the maximum photosynthetic capacity of terrestrial plants. It's totally true that Rubisco is a necessary component of understanding photosynthetic capacity, but what I want to promote to you today is the idea that it's not sufficient to understand this concept. Only focusing on Rubisco really has gotten us to a point where we have not been able to have a sufficient level of generality or efficiency or accuracy in explaining the relationship between photosynthesis and fluorescence. And one of the things we need to do moving forward is to broaden our conceptual model of photosynthetic capacity to go beyond Rubisco as being a key component of controlling the capacity of carbon metabolism, and also recognize there's another equally important enzyme that sets the capacity for the electron transport system. And that enzyme is the cytochrome B6F complex. This is a cartoon of the placement of the B6F complex within the electron transport chain. And the way that we know that the B6F complex is really important to photosynthetic capacity comes from our main optical measurements of photosynthesis. When you shine a light on a leaf, you see the increase in fluorescence coming from photosystem 2, coincident, which is indicating a reduction at photosystem 2, and you see that coincident with oxidation at photosystem 1. And that concerted upstream reduction and downstream oxidation tells you that there's a kinetic bottleneck in the middle of the chain. And that bottleneck occurs within the cytochrome B6F complex. What this means from a modeling perspective is that the B6F complex plays a role in the electron transport system that's analogous to the role that Rubisco plays in carbon metabolism. And what I want to sort of pitch to you today is the idea that by resolving the B6F complex in our models of photosynthesis, we really have three big opportunities. One of them is that we can get rid of the old Jmax parameter. Jmax is a really challenging parameter to deal with as an experimentalist, perhaps mostly because it has been measured under a range of different conditions. It doesn't fully separate the physiologic state of photosynthesis that's being evaluated from the underlying drivers of that state in leaf biochemistry. And we now have an opportunity to jettison JMAX by moving to a B6F-based model of the electron transport system. One of the cool things and unexpected things we found in developing this was that the B6F complex you could really think of as having been a missing boundary condition for understanding the coordination between the electron transport system and carbon metabolism. And once you bring that boundary condition correctly into the model, it becomes possible to quantify the major classes of regulation that coordinate energy supply and demand in a really simple and efficient way, including processes like state transitions and non-photochemical quenching and cyclic electron flow, and all of these important things. The third sort of part of the value proposition of this approach is that it gives us a simple way to move beyond calibrating photosynthesis models with measurements of fluorescence intensities. And it actually gives us a pathway to calibrate models and anchor them to fluorescence lifetime measurements. Sort of the third piece of this. So I have been saying these things in different forms to many of my colleagues for the last number of years, and some of them have bought into this idea. So these are folks who have been early adopters of the Johnson and Berry model and have implemented it in a range of different kinds of larger scale modeling frameworks, from what I would call classic land surface models to hybrid models or satellite simulators, to pure radiative transfer frameworks. You have seen earlier today some results from BESS, and are we going to call it TEX? I think we're going to call it TEX. And what I'd like to do next is to show you some results from this first class of models, first from ELM Fates and then from BEPS, that demonstrate different aspects of how it goes when you switch from the Farquhar scheme to the Johnson and Berry scheme for modeling photosynthesis and fluorescence. So this first example is a study that is currently in review. It was led by Julian Lamore under the supervision of Alastair Rogers at the Lawrence Berkeley National Labs. And what they did was to ask and answer a question that many people have asked me, which is, will it really matter to our analysis of leaf gas exchange if we use this new electron transport model as compared to the empirical models that have been part of the Farquhar framework? So what Julian and colleagues did was to compare the new framework against the Farquhar model in a standardized way against more than 1,000 light response curves representing more than 100 different C3 species. And they found that, indeed, most of the time, the new modeling framework achieved a better fit to the gas exchange data than a conventional implementation of the Farquhar model. And when I say that, I mean an implementation where the curvature parameter for the electron transport function is fixed, and the asymptote of the response is allowed to vary with the Jmax variable. The reason for the improved fit was basically that the curvature is more realistic in the new model. They were also, in this analysis, able to address the question of the generality of this framework across different plant functional types. The data they were comparing to included species from all of the major terrestrial biomes. Oh, we gotta go quick. And indeed, this result was consistent across all of them. They then implemented this the Johnson and Berry framework with this parameterization into the E3SM land model and found that just switching the model type gives about an 8% difference in global terrestrial GPP. And I want to note that this is about of the same magnitude as the signals that you were hearing about in Nick Parazu's talk. So this is a very small detail to the photosynthesis scheme, but that has a big effect globally. I'm going to quickly also say that there's an important new study coming out in global change biology that's been led by Yu Lu under the supervision of Yangwan Zhang and Xiaoying Zhang, who I think is here somewhere. I don't know if she can wave. And since I'm getting short on time, and I believe she's here somewhere, I will go ahead and move through this quickly just by saying this team has also evaluated the Johnson and Berry scheme in a different land surface model within BEPS against data at 16 tower sites, both eddy covariance and tower spectroscopy. And the new scheme works pretty well, particularly under the kinds of environmental conditions where you'd expect to see advantages. Low light for GPP, high light for SIF, and in particular, the improvement in the model's ability to simulate the SIF observations is really being driven by what happens in the sun leaves as compared to the shade leaves. So this will be out soon, and you can take the figures in more detail then. What I want to pivot to before I run out of time is the relationship between this and the work that my group has proposed for the FLEX-Calval AO. Essentially, what we're going to be doing is implementing the Johnson and Berry scheme within scope, and we're expecting that that will lead to improvements in modeling the relationship of photosynthesis and fluorescence, particularly in these first, what I'll call 2 stages of the Phi-P and Phi-F scatter plot. So you could call the first one light-limited photosynthesis, and the second one light-saturated. We also call them cytochrome B6F limited and Rubisco limited photosynthesis. And the hope is that that will provide an improved foundation for studying and trying to understand stage 3, the stress domain that's been highlighted by many of the speakers today. I had one more really important slide. Did I get cut off?
Maybe a technical issue?
There is. Okay. So in our project proposal, what we've put forward to do is to come up with an approach to quantify the maximum photosynthetic capacity of terrestrial plants by using FLEX mission products. We'll essentially use the implementation of the Johnson and Berry model within scope to perform inversions that are driven by the FLEX data. And then we're going to be attempting to compare the protein fields that we retrieve from those inversions to in situ measurements. The hope in the long term is that this will, if we can get it to work, that this will lay the foundation for development of new experimental science data products that resolve fields of Rubisco and cytochrome B6F in much the same way that the planned data products resolve fields of carotenoid and chlorophyll pigment pools. Okay.
Thank you. Questions, audience?
Yeah, it is a very nice talk, Jen. So one quick question. I found the difference between Farquhar model and your model was very severe in tropics. So why?
You said it was which direction in the tropics?
The difference between Farquhar model versus your model
on
in GPP.
The global plot.
Yeah, global map.
The reduction in the reduction in GPP is concentrated in the tropics
Yes.
because the magnitude of GPP is highest in the tropics. So this curvature change is affecting all of the biomes, but the magnitude impact that it has on the estimation of carbon fluxes in absolute terms is proportional to the gross, the GPP flux.
Okay.
Okay, how can you can use fluorescence lifetime data to calibrate the model? Can you develop or give more detail on this point?
On approaches for calibrating the parameters in the model? This is the question?
You say that there was a slide where you say that fluorescence lifetime can be used to calibrate the model. So would you develop or
yeah, for sure.
Give more details on this?
So what we've done with the published version of the model is to calibrate the full set of photochemical rate constants for photosystem 1 and photosystem 2 so that they respect fluorescence lifetime measurements that were made in vivo at room temperature. And that gives us a sort of anchor point for the defined maximum photochemical efficiency and fluorescence efficiency for photosystem 1, as well as a minimum and maximum of photochemical yield and fluorescence yield for photosystem 2, and then any state intermediate will land within those calibrated ranges. I want to say I think we have a good approach for doing this, and there's a lot of room to improve the actual numbers that we use for those calibrations from new experimental studies that use lifetime measurements in a broader range of species and with the correct kind of physiologic poising under the right environmental conditions.
Okay, thank you.
Wonderful, thank you very much, Jennifer. And our next speaker is Federico Grillini from the University of Copenhagen. Please, the floor is yours to tell us about how to link SIF to biogenic volatile organic components emissions.
Hello, thank you Marco for the introduction, and thank you all for being here. And I'm in the grim spot of being between the end of the program I'm in. And since we are in the bit of a different session, I would like to start a bit differently. So if you don't mind, maybe we can do a brief sensorial experiment where you could just close your eyes and imagine that in 20 minutes time about that, you're approaching the coffee break and you're taking this coffee mug to your mouth. Done. Experiment is over. You can open your eyes, and maybe I was able to trick your brains into smelling this guy, a guaiacol. Funny guy. This is a biogenic volatile organic compound, and who also emits this kind of compounds? Plants. And plants emit these compounds for various reasons. They emit them to communicate to each other, to attract pollinators, and they emit them in response to some attack by some eaters to send them away, and also they emit them in response to biotic and abiotic stresses. And why these compounds are interesting and important is because their feedback on the climate is not yet fully understood, and there is still studies going on and trying to understand if it's a positive feedback, so a positive loop of increasing warming or decreasing warming. In the first case, it could be that these volatile compounds could extend the lifetime of methane in the atmosphere, and so we know methane is a powerful greenhouse gas. In the other case, they can contribute to the formation of aerosols, which could contribute to the formation of clouds, and clouds can limit the amount of radiation reaching the surface, so they could limit a bit the warming. But these are all studies that are still going on, and I'm not the person to talk about that. But I'm the person to talk about why the Arctic is important, and we've seen it in some presentations already. The fact that it's a quite understudied and unexplored environment, but it is also the one that is warming up faster, much faster than the latitudes where we live. And this warming can lead to earlier snow melt onsets, which leads to an earlier start of the growing season, which combined with the long time of sunshine during the summer, this creates like a perfect combination for plants to proliferate and have higher vegetation productivity, BVOCs being one of these things being produced. And BVOCs, they have been recently linked to some quantities that we could measure with remote sensing, and especially SIF and PRI, with the explanation that they can share functional activations. And so there is a big advocating work that is trying to link the two, but not a lot of work has been done, especially with the remote sensing with classical approaches that we know. For example, there has been work in linking isoprene emissions with PRI. Yeah, we can see here the relationships and also the relationship here and how it is modeled with different complexities. And also there has been some work that has been done in trying to create some sort of hyperspectral isoprene indices where these are just like band math combination, and so trying to highlight what are the important bands to create a sort of index that could be used. And as well, there has been a recent work using leaf spectroscopy in the Amazon forest and linking isoprene emissions to the reflectance, but in this case, just looking at the importance of the wavelengths. But as you can see, there hasn't been really a work trying to connect SIF with the emissions of BVOCs, and that's where my group came in, coming up with some research questions. But for the sake of time in this talk, we really will focus only on the last one. So can we deploy SIF as a marker for these emissions? And the way that has been done, it's going to Greenland as a study site for the Arctic, for Arctic tundra, and gonna go to Kobbe Fjord, which is this beautiful fjord that is just southeast of Nuuk, the capital. Maybe you have heard about it in the last months. And in this site, in the summer of 2023, were established 12 plots where the vegetation is quite, there is a specific species that is dominant. And then in summer of 2024, so 1 year after, to give the vegetation some time to sort of adapt to the new condition with the frame, there has been conducted the field activity, so BVOC measurements and field spectroscopy. And these are also the species that are evaluated in the study, and these are very, very common vegetation in the area, and they're all low shrubs, so they could fit in these frames, and the height is really not taller than 20 centimeters. And for the BVC measurements, we use the setup developed by VOLT, the Volatile Center in the University of Copenhagen. And here you can see why having this steel frame is important, because we need to mount these. There are effectively oven bags where you can cook turkeys inside of it, but they've been sterilized for the use, and so you can create an enclosure of this and this chamber, and with hoses then you can push and pull air through a pump. And in the first phase we have a flushing where we just make a new airspace in the chamber for 15 minutes, and then in the next 30 minutes the sampling takes place, and these volatile compounds get trapped in these 10 tubes that are inserted at the start of the hose. And you can tell the times, 15 minutes, 30 minutes, it takes 45 minutes to do one sampling for one plot. So this is quite laborious work. Then you have all the conditions of flying to Greenland, having limited time. Then you have bad weather, to say a good word. And so this limits really the amount of data that we can collect. And also we cannot really revisit the plot and get continuous measurements because these are quite stressing sampling procedures, but at least they give us a really very good specificity of where we are. And then these 10x tubes are sent back to the lab once back in Copenhagen for gas chromatography, mass spectrometry. And then all these compounds, they get identified and then grouped into the main groups. And here we're going to just focus on the first three, which are isoprenoids. In our study, we also considered more, but we don't have time, and also have to show just the ones that are important for SIF, right? And this is the setup for field spectroscopy. And we used the FOX instrument, and yes, that is not a typo, it is a FOX instrument. And after talking with Andreas from JB Hyperspectral, we convened that we can define this as the cousin of FLOX, apparently. And it works exactly in the same way, but it just has the spectral resolution that is slightly, it's not as good as the fluorescence sensor of the FOX, the newly one, but it's still below 1 nanometers resolution, so it's still pretty good and allows us to calculate SIF. So we have here the setup, and here just for, you can see that these frames are quite reflective, but we also have some dark cloths. The inside of this is very dark, so we can wrap them around to limit the radiation, the unwanted radiation, at least. And then we also have our operator, Simon in this case, who we will lie down during the sampling to avoid, to have a uniform radiation captured by both the up-looking and the down-looking sensor. So what we get is something looks very familiar. We get the fluorescence peaks here, and these are just averages across the 5 visits that we did over the summer. And then from these we can get, of course, fluorescence, and then we can get spectral indices from the whole spectrum extension. These are just some of the results to show a bit the range of the values that we get for SIF, for example. And then we also calculate common indices like PRI, NDVI, EVI, and chlorophyll-related spectral indices as well. What we wanted to do then was just to have all these variables, because I forgot to mention that we also measured environmental variables like soil temperature, air temperature, and soil moisture, for example. And we pulled everything inside the PLSR model, ran backward sequential feature selection and the permutation importance to get a ranking of variables that are important into explaining specific BVC groups. And we done this individually for each group. And what it's quite evident is that a bunch of these spectral indices can be found at the top of the rankings. And what is also interesting for us is that SIF appears at the top of the ranking quite a lot. You can also consider that there are more variables just under here that just didn't make the cut. And what we built from that ranking is structural equation models to assess and explore the causality between all these variables. And these are made in a way that these relationships need to make sense. So in a way, we know that solar zenith angle can affect SIF, but we don't have the other way around. And each path here is a linear mixed effect model. And the way to read this, it's a bit complex, and I wasn't really used to work with this before this work. So we have like coefficients standardized that tells us a bit the power of the connection, but also we have the ranking 1, 2, 3, 4, 5 in terms of what are the most significant connections within this model. And so here we can explore, and what we were happy to see in this case was that SIF can maybe be used as a marker for isoprene. Then we moved for example, the other groups, and we didn't have SIF up there in the ranking, and we had EVI, for example, for monoterpenes, and we see here the significant relationship. And same we see PRI, which was, if you remember at the start, was one of those indices that was advocated to be important for, it was isoprene in that case, but we see here sesquiterpenes, which is another compound. But then we weren't really sad to not have SIF here, because we were happy of just having anything that we could measure remotely, and we could connect to the BVOCs. But then we still forced the models to include SIF, because this is what you can do with this kind of models, and it came up that the significant relationship were still present also for monoterpenes and sesquiterpenes. I'm just going to move a little bit quicker. For the total BVOC model, with total BVOC just the sum of all the compounds, so not only the isoprenoids that I showed you, but also all the other ones that have been considered, we still see a relationship with SIF, which can be explained by the fact that we saw it for the individual compounds earlier, and if you look here, those are quite the ones that are quite prominent in the blend, so it makes sense to see that. And yeah, to conclude, what we were happy to see was in a way that we can use quantities that can be remotely sensed, like SIF in this case, but also other indices for these compounds. And the question that we might ask ourselves is, can these indices be deployed on their own? And the answer that I'm giving you is absolutely no. We still need the classical factors like temperature and probably also PAR and Liefera index if we're able to calculate that from the field. And then there is, of course, the outlook on how we move from here and then scale up, but we have had very nice talks on that, and I'm definitely not the person to talk about that. And what I can just say that the main outlook for this is maybe, I don't know, to thank you as a community for having all these efforts to send FLEX up in orbit, because maybe you didn't think about these as an application at the start, or maybe you did, but maybe you don't know what is the next application that can benefit from having a product like SIF, so from space. So it's just great. And yeah, that was it. I want to thank all my authors and the field assistants and the support, of course. So yeah, thank you.
Thank you very much. Any questions? I would.
Thanks for your talk. Can you go back to slide 13 maybe? Where you listed the importance. And for the total BVOC, you see that the SIF efficiency is the lowest at the bottom in the relative importance. And you have the FCVI and the NRVH2 in the top ranked. So does it actually make sense to use SIF, or would any hyperspectral data already be sufficient?
So that's a good question. The fact is that here we saw this importance of FCVI and not so much from the efficiency, which is yes, last here, but this is also just a representation of the top 15. There were also more. So yeah, it's last, but it's top 15. So it's not as last in a way. Yeah, if we then look at the model that came up from it, we then see that FCVI doesn't really have an effect. At least we use these models to sort of have more interpretable results, because the ranking that I showed you comes from a PLSR, so it's a linear combination of all the variables. But then when we pull it back in here, that significant sort of disappear. Our goal was to sort of go here to the structural equation models, but we had way too many variables to just start to unless we just had a clear idea of where to go. So we ran that ranking before.
Yeah. Okay. Thank you very much.
Would be really curious to understand how do you say address or understand. Demonstrate the causality that you mentioned in this approach compared to, say, a purely statistical one, mostly from my understanding of how you build these models.
Well, the causality, we sort of, I guess, explain it a bit a posteriori after seeing the results, because the way we built the model is only with, let's say, logical relationships. So the way we build these paths, we can only assume that SIF can have, for example, in this case, can be influenced by the Leaf-Air index, which in this case wasn't really a Leaf-Air index, but was a proxy for Leaf-Air index calculated with some band math. So already it could be a little bit of a stretch to have this connection if you want, but so the Leaf-Air index would be a one. We could argue that maybe there is a connection from temperature, and we can hypothesize that, and that the species makes. But then, yeah, the causality in our case, we can maybe say that SIF in this case has an effect on the total BVOCs, but through sort of an effect from temperature as well. And maybe from Leaf-Air index, but if this was the real Leaf-Air index. Yeah.
Thank you. Okay, very good. So we reached the end of the, let's say, of the stream of from the talks to the audience. We would still have some minutes to hear from you in terms of, let's say, things which we might have not captured, either on the purely aquatic side or on other elements which, let's say, could be sort of innovative science products which are enabled by the availability of FLEX data. Of course, I mean, this is a chance to bring this up. We have also this virtual Q&A space to do that, and also we will have 2 discussion sessions. One at the end of the morning sessions tomorrow, mostly a recap of the highlights, but hopefully also some inputs from you. And then on Friday morning, the last 2.5 hours basically of the workshop dedicated to that. But in case there is any inputs or ideas or things which you have seen in the past which you think have not been particularly discussed or represented in these presentations, it's your chance. Yes?
Regarding the coastal areas research, is there any plan to include research on seagrass areas and maybe to protect them?
Yes, so it's something we are expecting to do this year, actually, to get into shallow waters, to get the fluorescent signal from the flocks. But I would say that it's very basic science. We have no algorithm to apply to FLEX directly, but I think it's very promising to see how fluorescence will decrease with the depth as well. So it will be between bathymetry and bottom habitats monitoring. But the problem we have right now, it's a special resolution. 300 meters sometimes is too coarse to make this kind of mapping.
Thanks. I'm a little bit inspired by the last talk. And what I understand is you try to measure VOCs, which are an important compound in the atmosphere. However, they are so low concentration in the atmosphere that you cannot detect them directly. So you're looking for proxies, which could be used as an indicator where you have the VOC emissions. Did you, or maybe also the other ones, think a little bit one step further, not using the SIF signal as a proxy, but really going one step back to use the high resolution, the spectrally high resolution FLEX data, which we have, atmospherically corrected to the top of canopy level. So we have spectral data with 0.3 nanometer spectral resolution. Is there a chance maybe to detect, in your case, VOCs directly, because you may be just lucky that you have a very specific absorption reflection feature in the spectral range?
Yeah, that's I think what we sort of set up at the start with, and I think that's what also one group started doing to sort of try to develop this isoprene hyperspectral index. I haven't evaluated that, but I guess yes, it could be very nice, and I guess the limitation of the 300 meters for Flex would be a bit challenging because plants emit VVCs very differently. So even if you have, unless you have a very homogeneous diversity, which is not a diversity anymore, it will be very, very hard to sort of connect those emission factors. So it would be challenging with the resolution, I think. But yeah.
Yeah, I'm Denis Macedounas from Wageningen University. I wanted to mention that one of the things that I would like to work on for FLEX as a global coverage, or mostly global coverage, hyperspectral mission is to improve land cover maps at the global scale. So for that I also have a poster. So if you're interested in that, then please come to it and discuss a bit more.
Thank you.
In the back here. So yeah, it's my understanding that you're going to provide the land surface temperature as part of the level 2C vegetation product suite. And I assume that's over land. Wondering if there are any plans to include that with the coastal water data set, I guess the red SIF, if that's going to have land surface temperature, which could be a valuable kind of indicator of the driver of harmful algal blooms or changes in water quality, that kind of thing.
Yes, in principle, my understanding is that for every level 1B, Flex level 1B product and level 1C product, and the full suite of, say, of complementary information from Sentinel-3 will be provided. So when Tristan showed that mask before, so we have a sort of baseline acquisition mask, which is over all emerged land and coastal areas up to 370 kilometers. And with this Flex for Water study, now we are somehow revisiting the chance for additional aquatic acquisitions, but this inland and coastal waters are included in the baseline acquisition. So basically each and every product will be produced like that. That's maybe for someone from the disk to confirm. I see Roberto there, but that's my understanding.
Yes, well, Flex, it's a fantastic mission, probably the best mission we have for capturing the photosynthetic dynamics. But at the end, the main bottleneck still remains the low temperature resolution, right, for capturing dynamics. So here the question is, are there already any plans to think of higher level products that are fusion products? So for instance, fusion with RoboSIF or other kind of SIF missions that enhance in this way the temporal resolution.
Short answer is yes. The idea is not boycotting. No, joking. The idea is there. There is no, to my knowledge, there is no structured activity already in place for that. But yeah, the idea is definitely there. And I think it's also a little bit up to the community to come up with clever proposals how to do that, right? And so there are multiple calls or multiple frameworks in which this could be implemented, not necessarily as a directly Flex product, right? There is Marine is not here at this very moment, but in the CCI vegetation, which is an is a postgraduate study. Now CIF is being considered. Christian knows probably better than me because he's in this project, and so I think there are multiple frameworks in which this could happen. But yeah, certainly the idea is not forgotten. Okay. Thank you very much for this afternoon. Although short, I think it's been quite interesting. Now we have a poster session, and then just as a reminder, from 7 p.m. For those of you who are joining the dinner, the open bar will be open, and then from 7:30 also the buffet will be available. So. Looking forward to see you through the posters and in the room for the next couple of hours, and then from 7 p.m. in the Hotel Ameron. Thank you very much again.